Hello. Tonight we'll spend some time exploring a civilization that has existed for 100 million years. One that has outlasted empires, ice ages, and mass extinctions, and that continues right now beneath your feet, inside the walls of your home and under the bark of every tree in your neighborhood. They have no president, no central command, no written law, and yet they build cities with climate control, run farms that have operated for 50 million years, and wage wars with strategies that military theorists have studied in earnest. They are, by almost any measure, the most successful land animals in the history of this planet. Tonight, we are going to take them seriously, not as pests, not as a curiosity at a picnic table, but as what they actually are, a form of civilization so alien to our own that understanding it requires us to rethink what we mean by intelligence, society, and even life itself. We will move slowly. We will look closely. And by the end of this, the next time you see a single ant crossing a sidewalk, you will not see the same thing you saw before. An ant can carry between 10 and 50 times its own body weight, depending on the species. That number gets repeated often. What gets skipped is what it actually means at the level of muscle and physics. An ant's muscles occupy a far greater proportion of its body than human muscles do, relative to our frame. Somewhere between 1/4 and 1/3 of total body volume is dedicated to locomotion and grip. Because the ant is small, the cross-sectional area of its muscles relative to its mass remains highly favorable. Strength does not scale linearly with size. It scales with the cross-section of muscle fiber, while mass scales with volume. A larger animal carries proportionally more of itself just to exist. The ant, by staying small, sidesteps that burden entirely. Think of it this way: if carrying capacity scaled the way ant muscle does, a creature the size of a golden retriever could carry a compact car in its mouth without breaking stride. That thought experiment is not whimsy, it is the actual ratio translated into a size a person can picture. Scientists still debate exactly where the upper limit falls for the strongest species, but the biomechanical principle is not in dispute. Smallness in this case is not a limitation. It is an engineering solution. The ant wears its skeleton on the outside. That sentence sounds simple until you consider what it demands of a material. The exoskeleton, the hard outer shell, called the cuticle, must be rigid enough to protect internal organs, flexible enough to allow movement at dozens of joints, light enough not to impede the ant's remarkable strength to weight, in ratio, and waterproof enough to prevent desiccation in dry environments. It is made primarily of chitin, a long-chain polymer, also found in fungal cell walls and crustacean shells, layered with proteins that can be hardened or kept pliable depending on the body segment. The joints between plates are membranous, softer zones that allow bending without cracking. In some species, the cuticle contains wax layers that seal moisture in with the efficiency of industrial sealant. What makes this material genuinely remarkable is that the ant grows it from within and sheds it entirely when molting through larval stages, then rebuilds it from scratch. There is no repair mechanism for a crack in the adult cuticle. The ant lives within its shell the way a sailor lives on a ship, maintaining what can be maintained and accepting that the hull once built is what it is. An ant's brain contains roughly 250,000 neurons. A human brain holds approximately 86 billion. By raw count, the comparison seems dismissive. But neuron count alone is a poor measure of what a brain can do, and myrmecologists, scientists who study ants, have increasingly shifted toward looking at neuron density and the ratio of brain mass to body mass. By that second measure, some ant species have brains that occupy up to 15% of their body volume. In a human, the equivalent proportion would mean a brain the size of a small child. That density does not produce the same kind of thinking a human does. The architecture is entirely different, but it does produce something navigational. Memory, chemical language, processing, social recognition, and coordinated behavior that has puzzled researchers for generations. The Saharan silver ant, for instance, can navigate home across a featureless desert after a foraging run that covers hundreds of body lengths in shifting sand. It does this using a brain smaller than the period at the end of this sentence. Scientists still debate exactly how much of this is learned versus hardwired, but the performance itself is not in question. The brain is tiny, what it does with that size is not. The mandible is the ant's primary interface with the world, and calling it a jaw is like calling a Swiss Army knife a blade. Depending on the species, mandibles are used to cut, carry, fight, signal, dig, build, groom, feed larvae, and in at least one documented case, to generate sound by scraping against a specialized body ridge. Their shape is one of the most reliable ways to identify an ant's ecological role. Leafcutter ants carry serrated mandibles shaped like miniature saw blades, curved with fine teeth along the inner edge, optimized for slicing through vegetation many times tougher than the ant itself. Army ants carry long, curved, fang-like mandibles, built for gripping prey rather than cutting. Trapjaw ants carry mandibles that open to 180 degrees and close faster than almost any other biological mechanism on Earth. The same basic structure, two hinged appendages at the front of the head, has been refined by evolution into tools so varied that comparing a leafcutter's mandible to a trapjaw's is a bit like comparing a bread knife to a different bear trap. What they share is precision. Ants do not have hands. The mandible is everything, and evolution has treated it accordingly. Ant eyes do not work the way human eyes do, and the difference is more interesting than the limitation. Most ant species have compound eyes, clusters of simple lenses called ommatidia, that produce a mosaic image with lower resolution than a human eye, but a much wider field of view. What makes this more than a footnote is that many ant species can detect polarized light waves oscillating in a single plane, which the sky produces in consistent patterns relative to the sun's position. To an ant sensitive to polarization, the sky is a navigation map. Even on an overcast day, polarized light penetrates cloud cover in detectable patterns, giving the ant a compass that does not depend on seeing the sun directly. Desert ants of the genus Cataglyphis, the same animals mentioned for their remarkable navigation, use this system with a precision that has been confirmed in controlled experiments involving polarizing filters placed over outdoor foraging. Arenas. When researchers rotated the filter and changed the apparent polarization angle, the ants rotated their homeward paths by the same degree. Something in their visual system was reading the sky the way a sailor reads a chart. It is a reminder that perception does not require similarity to ours in order to be sophisticated. An ant's antenna is not simply a feeler. It is the primary organ through which an ant understands the world, and its capabilities have no clean human equivalent. Each antenna has two segments, a long basal segment called the scape, and a multi-jointed flagellum that can bend, sweep, and probe with considerable independence. The surface of the flagellum is covered in sensilla, microscopic sensory hairs, pegs and plates, each tuned to detect different kinds of information. Some sensilla are mechanoreceptors sensitive to air movement and surface texture. Others are chemoreceptors detecting airborne and contact chemicals. Still others respond to humidity and temperature gradients. An ant tapping its antennae against a nestmate is not simply touching it, is reading a chemical signature, checking nestmate identity, and potentially exchanging information about food sources or colony status, all in a gesture that takes less than a second. There are ant species whose antennae are so sensitive to humidity that they can detect the moisture gradient above an underground water source and adjust their tunnel direction accordingly. The antenna is in some meaningful sense a nose, a hand, a thermometer, and a social reader, all built into a structure thinner than a human hair. A dog's sense of smell is often used as the benchmark for olfactory excess, the standard against which other animals' chemical perception is measured. A dog has roughly 300 million olfactory receptors. A human has about 6 million. An ant, depending on the species, carries between 400 and 700 distinct chemoreceptor gene types, which may not sound like a large number until you consider that the entire genome of most ant species contains around 17,000 genes, meaning that somewhere between 2 and 4% of the ant's total genetic real estate is dedicated exclusively to smell. The fire ant genome, sequenced in 2011, revealed over 400 olfactory receptor genes. Many of these are tuned not to food, but to social information, nestmate recognition, queen status, alarm signals, trail markers, and chemical signatures that distinguish friend from intruder. In a colony of millions, every individual must be chemically legible. The result is an olfactory vocabulary of remarkable complexity, one that researchers are still in the early stages of decoding. If you have ever watched an ant pause, antenna tip to the ground, then change direction with apparent certainty, you have watched an animal reading a sentence written in molecules. It simply requires a different kind of literacy to appreciate. A queen ant's body undergoes one of the most dramatic physical transformations in the insect world, and it happens once irreversibly in a single afternoon. Before her mating flight, a young queen carries large functional wings attached to powerful flight muscles packed into her thorax. Those muscles are metabolically expensive to maintain and serve exactly one purpose: getting her airborne long enough to mate. After the flight, once she lands and begins founding a colony, she does something that has no parallel in vertebrate biology. She tears her own wings off, not by accident, but deliberately using her legs and mandibles in a process called dilation. Then over the following weeks, her body reabsorbs the flight muscles entirely, converting that protein mass into nutrients to feed her first clutch of eggs. Her abdomen expands. Her ovaries, initially modest, develop into structures capable of producing millions of eggs over a lifetime that can exceed 20 years in some species. The queen, who lands in a crack in a sidewalk and seals herself in, is physically not the same animal that launched from a tree branch that morning. She has already begun to become something else, a living factory, a dynasty compressed into a single body. Inside a single ant colony, every worker shares an identical genome with every other worker. They are in the strictest genetic sense the same animal. And yet a minor worker and a major worker in a leafcutter colony look so different that early naturalists occasionally classified them as separate species. Minor workers are small, narrow-headed, and built for carrying leaf fragments and tending fungal gardens. Major workers, sometimes called soldiers, carry heads so disproportionately large that the head itself can account for more than half the animal's total body mass. The mechanism behind this divergence is not genetic mutation but epigenetic regulation. The same DNA is read differently depending on nutritional signals and hormonal exposure during larval development. Small differences in diet during a critical developmental window can shunt a larva toward a completely different body plan. Researchers have confirmed this experimentally by altering the juvenile hormone levels in developing larvae and watching the resulting caste composition shift accordingly. The colony does not simply grow workers, it manufactures them to specification, adjusting the production ratio in response to current needs. When soldiers are lost to a predator attack, the colony produces more. When foraging demand rises, minor worker production increases. The genome is a blueprint. The colony decides what to build. The soldier caste in many ant species carries a head so large relative to body size that it presents a genuine structural engineering challenge. In some Phytol species, a genus so numerous that the biologist E.O. Wilson once called it the dominant genus of ants, the major worker's head is roughly 3 times the width of its thorax. That proportion in a human would mean a head wider than the shoulders. The head's size is almost entirely accounted for by jaw-closing muscles, thick layered adductor muscles, that generate crushing force far exceeding what the minor worker's slender mandibles could produce. In certain species of turtle ants, the soldier's head is not merely large, it is flat and disc-shaped, precisely sized to plug the nest entrance like a living door. The soldier positions itself in the tunnel, opening head flush with the colony's threshold, and holds. To enter, a returning nestmate must present the correct chemical signal, tapping the soldier's head with its antennae. The soldier then steps aside. An intruder receives no such courtesy. The head, in that case, is simultaneously a key reader, a door, and a lock, and the ant did not learn this behavior. It grew into it. Between the thorax and the abdomen of every ant lies a narrow constriction, 1 or 2 small nodes depending on the species, called the petiole. In some ant families, a second node called the post-petiole follows it, giving the ant its characteristic pinched silhouette. This structure is not decorative. The petiole solves a mechanical problem that arises from the ant's remarkable flexibility: how to allow the abdomen to swing in any direction, forward, backward, sideways, while maintaining structural connection to the thorax. The flexible nodes act as universal joints, giving the ant the ability to curve its abdomen beneath its body to sting forward or above its body to spray acid at a target directly ahead. Without the petiole, the ant's abdomen would be fixed in a single plane and its defensive chemistry would be largely wasted. The petiole also plays a role in species identification. The number of nodes, their shape, and their proportions are among the first features a myrmecologist examines when identifying an unknown ant. In the taxonomy of ants, the petiole is as diagnostic as a fingerprint. It is a small structure carrying considerable responsibility which, on reflection, describes a great deal of what makes ant anatomy worth studying. Ants do not breathe. Not in any way that resembles the word as most people use it. There are no lungs, no diaphragm, no rhythm of inhalation and exhalation. Instead, oxygen enters and carbon dioxide exits through a series of small openings along the sides of the body called spiracles, typically one pair per body segment. From each spiracle, air travels inward through a branching network of tubes called tracheae, which subdivide into increasingly fine channels called tracheoles that reach directly to individual cells. Gas exchange happens at the cellular level without the intermediary of blood carrying oxygen molecules the way mammalian circulatory systems do. This system is extraordinarily efficient at small scales, which is part of why the basic insect body plan has remained largely unchanged for hundreds of millions of years. It does, however, impose a size, ceiling diffusion, through tracheal tubes becomes inadequate beyond a certain body volume, which is one reason insects never grew as large as vertebrates, after atmospheric oxygen levels normalized following the Carboniferous period. An ant breathes the way a city distributes water through pipes, laid throughout the structure, reaching every point without needing a central pump. Where human blood moves in a closed loop, veins carrying it back to the heart arteries pushing it out, ant circulation is an open system. The fluid inside an ant's body called hemolymph is not contained within vessels for most of its journey. A simple tubular heart runs along the back of the ant's abdomen, pumping hemolymph forward through the body cavity, where it bathes the organs directly before slowly making its way back. Hemolymph is typically pale yellow or colorless. Ants have no hemoglobin, the protein that makes vertebrate blood red. Instead of carrying oxygen, the hemolymph transports nutrients, hormones, and immune cells called hemocytes. These hemocytes serve a function analogous to white blood cells, identifying and engulfing pathogens. In some species, they also carry out a more dramatic immune response. Certain ant species contain specialized cells that can rupture intentionally when the ant is severely threatened, releasing toxic or sticky compounds that immobilize. To attackers, this behavior, called autothysis, and found most dramatically in some Malaysian Camponotus species, means the ant's circulatory system serves as a last resort as a weapon. The body becomes the ammunition. Scientists still debate exactly how voluntary this response is, and whether voluntary is even the right word to apply. The difference in lifespan between a worker ant and a queen of the same species is one of the most extreme within species contrasts in the animal kingdom. Worker ants in most species live between a few weeks and a few months. The queen of the same colony may live 20 years or more. In Lasius niger, the common black garden ant found across Europe and North America, queens have been documented living for nearly 30 years in laboratory conditions, a record for any insect. This difference is not primarily genetic, since workers and queens share identical DNA. It emerges from the radically different physiological demands placed on each body type. Workers forage, fight, carry, and navigate. They are exposed to the full danger of the outside world. Queens, once established, rarely leave the nest and dedicate their biology almost entirely to reproduction. There is also evidence that queen-specific hormonal environments suppress some aging pathways that operate normally in workers. Think of the lifespan gap this way. The worker is a sprinter burning through her body at full speed, while the queen runs the same race at a fraction of the pace, over a course that lasts as long as some human childhoods. The genetic instructions are the same. The life they produce is almost unrecognizable. A leafcutter queen atta species found across Central and South America can produce somewhere between 150 million and 200 million eggs over the course of her lifetime. That number is large enough to be meaningless without translation. Laid end-to-end, the eggs from a single queen's reproductive career would stretch approximately the distance from New York City to Chicago. Every one of those eggs was fertilized using sperm collected during a single mating flight that lasted at most a few hours. The queen stores that sperm in a specialized organ called the Spermatheca and releases it in controlled quantities across decades, fertilizing each egg individually as it passes through her reproductive tract. Unfertilized eggs, by contrast, develop into males, a system called haplodiploidy, shared with bees and wasps, in which the queen controls the sex of her offspring through the simple act of whether or not to open a valve. The colony's gender ratio is not an accident of biology, but a managed decision adjusted in response to seasonal conditions, colony size, and resource availability. One animal, one afternoon of mating, and then 2 decades of population control exercised from a sealed chamber beneath the soil. The logistics of it are on reflection staggering. The Spermatheca, the queen's sperm storage organ, is one of the more quietly extraordinary structures in the natural world. In a mated queen, it contains sperm collected from multiple males during the nuptial flight, kept viable at body temperature, without refrigeration, for the entire duration of the queen's reproductive life. In atta leafcutter queens, that means sperm stored for up to 20 years continues to fertilize eggs successfully. For comparison, human sperm stored under clinical conditions, frozen in liquid nitrogen in a medical facility, begins to decline in viability after a decade, even under optimal conditions. The queen's Spermatheca maintains sperm at room temperature inside a living body with no external technology, and loses negligible viability over the same time frame. Researchers have studied the biochemical environment inside the Spermatheca and found a precise combination of antioxidants and low oxygen conditions that suppress cellular aging in the stored cells. The mechanism is not fully understood. Some researchers believe the Spermatheca actively monitors and culls deteriorating sperm. Others believe the storage medium itself is the key. What is not debated is the outcome: a structure smaller than a grain of sand, functioning as a 20-year biological archive, with a reliability that no human technology has yet matched at equivalent scale. When floodwaters rise in the lowlands of the American South, fire ant colonies do not drown. They assemble. Within minutes of sensing rising water, a fire ant colony can form a living raft, workers linking legs and mandibles into an interlocking mat that can support the entire colony, including the queen brood and food stores on the water's surface. The raft is waterproof not because of any coating applied to it, but because the ants trap air bubbles between their hydrophobic bodies, creating a layer of air that provides buoyancy. Tested in laboratory conditions, these rafts can remain afloat for weeks. The ants on the bottom layer are submerged but do not drown. They breathe through the air layer, held against their bodies by surface tension. The structure is self-healing. If a section is damaged, workers on the edges pull inward and close the gap. It moves directionally, drifting towards solid ground, with workers on the leading edge using their antennae to detect substrate. What makes this behavior particularly remarkable is that no individual ant understands what a raft is. No signal is given to begin construction. The behavior emerges from simple rules: move toward nestmates, grip what you touch, do not release, and the result is a watercraft assembled by animals that have never seen water before and will never build another one if the flood does not return. The trapjaw ant, several species across the genera Odontomicus and Mystrium, holds the record for the fastest self-powered movement of any biological appendage ever recorded. The mandibles of Odontomicus boweri open to approximately 180 degrees, locked in place by a latch mechanism analogous to a cocked spring, then close at speeds measured between 126 and 230 kilometers per hour, with peak acceleration forces exceeding 100,000 times the force of gravity. The strike lasts between 0.13 and 0.33 milliseconds, far faster than any nerve signal could initiate and terminate a muscle contraction. The speed is achieved not by faster muscles, but by an energy storage system. Muscles slowly load tension into a latch structure, and when a trigger hair is touched, the latch releases, and converts stored elastic energy directly into kinetic motion. The mandible is not swung, it is launched. This mechanism is used for prey capture, but also for escape. A trapjaw ant can aim its mandible strike at the ground beneath itself, launching its own body upward and away from a predator with enough force to travel several body lengths through the air. It uses its jaw to jump. Scientists still debate whether this secondary use developed as an adaptation or is simply a byproduct of having the fastest strike in the animal kingdom. The Schmidt Sting Pain Index is an entomologist's attempt to impose order on a deeply subjective experience, being stung by various insects, by assigning each sting a score from 1 to 4. Most familiar stings cluster at the lower end. The honeybee sits at a 2. The bullet ant Paraponera clavata, native to the rainforests of Central and South America, occupies its own category at the top, a 4+ described in the index, with language that reads less like science and more like testimony. The pain has been compared to being shot, which is where the common name originates, and it persists unrelenting for approximately 24 hours after a single sting. The venom contains a peptide called ponerotoxin, which disrupts sodium ion channels in nerve cells, causing them to fire continuously and uncontrollably. The result is not merely local pain, but systemic neurological disruption, trembling, nausea, and temporary paralysis of the affected limb have all been documented. And yet, among the Satare Mawe, people of the Brazilian Amazon, young men wear gloves woven with hundreds of sedated bullet ants as part of an initiation ritual. They must wear them for 10 minutes. The ants wake up. This is repeated 20 times before the ritual is complete. The ants, for their part, are simply defending themselves. They have no concept of ceremony. Weaver ants, Oecophila species found across Africa and Southeast Asia, construct their nests from living leaves stitched together with silk. What makes this unusual is where the silk comes from, not from the adults, but from their own larvae. The adult workers carry larvae in their mandibles and move them back and forth between leaf edges, stimulating the larvae to release silk from their labial glands, the same glands that in other ant species produce cocoon material. The larvae do not spin independently. They are used as living shuttles, their bodies squeezed gently to encourage silk output. A single nest can contain hundreds of leaf chambers bound together across several square meters of canopy, each seam the product of hundreds of larvae working in coordinated sequence directed by adults. The silk is strong enough to hold fresh leaves under tension for months. When the nest is damaged, workers repair it using the same method. There is something worth pausing on here. The adults in this system have evolved to use their own young, not merely as a next generation, but as a construction material, and the larvae, for their part, appear to suffer no lasting harm from the process. The silk glands regenerate. The larvae pupate, mature, and eventually carry their own larvae in the same way. Army ants, primarily Eciton burchellii in the Americas, do not build nests. They are the nest. When an army ant colony stops moving at the end of a day's raid, workers begin linking their bodies together, legs hooking into legs, forming chains that thicken into curtains, curtains that layer into walls, walls that enclose a central chamber where the queen and brood are suspended, insulated, and protected. This structure, called a bivouac, can contain 100,000 to 700,000 individual ants, assembled in under an hour, maintained through the night by the living tension of interlocked bodies, and disassembled at dawn when the colony resumes its march. The interior of the bivouac maintains a temperature several degrees warmer than the surrounding air, not through any heating element, but through the collective metabolic output of hundreds of thousands of bodies pressed together. The brood develops faster inside the bivouac than it would in ambient conditions. The ants regulate this temperature by adjusting the density of the outer walls, pulling tighter when it grows cold, loosening slightly when the interior warms too much. The bivouac breathes. It thermoregulates. It is, in the most literal sense, a living building, and every brick in it is also a citizen. The Saharan silver ant Cataglyphis bombicina lives in one of the most hostile thermal environments on Earth, and has evolved a solution so elegant that materials scientists have spent years attempting to replicate it. These ants forage on the surface of the Sahara Desert during the midday window, when surface temperatures exceed 70 degrees Celsius, hot enough to kill most insects within seconds of contact. Their triangular silver-coated hairs are the key. Unlike the round hairs found on most insects, the silver ant's hairs have a triangular cross-section that reflects sunlight across the entire visible spectrum, while simultaneously radiating heat away from the body in the infrared range, a passive cooling system operating on two different wavelengths at once. The result is a body temperature several degrees cooler than an uncoated ant of the same size would achieve in identical conditions. The hairs were studied by a team of researchers who published findings suggesting the reflective geometry could inspire new designs for passive cooling materials in architecture and textiles. The ant does not know it is doing any of this. It simply moves at speeds reaching 1 meter per second, among the fastest recorded for any insect relative to body size, covering ground quickly enough that heat accumulation never reaches fatal levels. Speed in this case is also a thermal strategy. The Dracula ant Mystrium camillii, found in parts of Africa, Asia and Australia, holds a speed record of its own, and the name comes from a feeding behavior that is stranger than its biology. Adults cannot digest solid food directly. Their internal anatomy is shaped entirely around liquid nutrition. To feed, adult workers make small wounds in their own larvae and consume the hemolymph, the larval body fluid that seeps out. The larvae survive this repeatedly, and the practice appears to be a fixed feature of colony nutrition rather than a stress behavior. It is the feeding method, not the jaw speed, that earned the common name. The jaw speed, however, is where the physics becomes genuinely startling. Mystrium camillii produces the fastest known animal movement ever, recorded a snap of the mandibles, measured at 90 meters per second, with acceleration forces exceeding 300 kilometers per hour in under a millisecond. This surpasses even the trap-jaw ant. The mechanism is a form of what physicists call a snap-through buckling instability. The mandible is pressed under increasing tension until it passes a geometric tipping point at which the stored energy releases instantaneously. The ant does not swing its jaw. The jaw snaps itself. The ant merely sets the conditions. Several species of arboreal ants, most studied among them, Cephalotes atratus, a canopy-dwelling species in South American rainforests, can glide, not fly, not fall. Glide-controlled, directed, aerodynamically managed descent from canopy height back toward the tree trunk, executed by a wingless insect using nothing but body posture and limb position. When dislodged from a branch by wind, predator strike, or experimental flicking by researchers, these ants spread their hind legs, orient their bodies with the head angled upward, and curve into a controlled trajectory that brings them back to the tree trunk rather than to the find forest floor. The forest floor in a tropical rainforest is ecologically distant from the canopy, a different environment with different predators and almost no route back up. Falling straight down is functionally fatal for a canopy ant. The glide is a survival mechanism. Researchers confirmed this by painting white stripes on tree trunks and filming ants dropped from heights. The ants consistently curved back toward the pale stripe, suggesting they visually target the tree during descent. Some species can return to the same tree from falls of more than 20 meters. The behavior requires no wings, no instruction, and no prior experience of falling. It appears to be present from the first moment the ant encounters open air. The smallest known ant in the world is Kerbara atomus, a species found in Sri Lanka, with workers measuring approximately 0.8 millimeters in length, small enough to walk through the eye of a standard sewing needle with room to spare. The largest is the driver ant queen Doralus, whose reproductive females can reach 52 millimeters, nearly the length of a human thumb joint. Between those two extremes lies a size range that, scaled to human proportions, would span the distance between a house cat and a draft horse. Both extremes represent functional, successful colony organisms. Both have persisted for millions of years without meaningful modification to their basic body plan. The smallest ants tend to occupy highly specialized ecological niches, living inside the nests of larger ant species as parasites or opportunists, where their size is the primary advantage. The largest tend to be colony founders or reproductive specialists whose body mass reflects the enormous energetic investment of founding a new colony from scratch. What unites them? Across 50-fold differences in body length and 1,000-fold differences in mass is the same fundamental architecture. 6 legs, 2 antennae, a petiole compound eyes, and a colony waiting to be built or maintained. The template is ancient. The variation built upon it is among the widest in the insect world. No ant in a colony of 1 million is in charge. No ant has a map of the nest. No ant knows how many foragers are currently outside, how much food remains in storage, or whether the colony is growing or contracting. And yet the colony responds to all of these conditions with apparent precision, redirecting foragers when a food source is exhausted, sealing tunnels when rain threatens flooding, increasing soldier production after a predator attack. This is what researchers mean when they describe ant colonies as systems of distributed cognition intelligence that exists not inside any individual unit but in the pattern of interactions between units. Each ant follows a small set of local rules, respond to this chemical signal, this way, move toward this concentration, retreat from that one, and the sum of millions of these local decisions produces behavior that looks from the outside like planning. The colony is not smart the way a person is smart. It is smart the way a market is smart, or the way a city's traffic flow optimizes itself without any single driver knowing the whole network. Scientists debate whether intelligence is even the correct word for this phenomenon, or whether applying it risks importing assumptions that obscure more than they reveal. What is not debated is the outcome: a system with no central controller that consistently outperforms any individual's capacity to manage it. Stigmagy is the principle by which ants and several other social insects coordinate complex collective behavior without direct communication between individuals. The word comes from the Greek for to incite work, and the mechanism is straightforward. One ant's action modifies the environment in a way that influences the next ant's action, which further modifies the environment, producing a self-amplifying cascade that generates large-scale structure from simple local responses. When an ant deposits a pheromone trail, it is not instructing other ants. It is modifying a shared medium. Other ants respond to the modification, reinforcing it if they follow the same path, allowing it to fade if they do not. The trail that survives is the one that was followed most, which tends to be the shortest or most productive path because ants traveling shorter routes return faster and deposit reinforcing pheromone more frequently. No ant planned the shortest path. The shortest path selected itself through the accumulated behavior of individuals who knew nothing about path optimization. Engineers studying this process have applied it directly to telecommunications network routing, where the same principle reinforced successful paths, allow unsuccessful ones to decay, now underpins some of the algorithms managing internet traffic. The ant colony solved a version of the traveling salesman problem millions of years before the problem had a name. When researchers present an ant colony with a foraging challenge, 2 bridges of different lengths connecting the nest to a food source, the colony consistently concentrates traffic on the shorter bridge, within a predictable time frame even. When both bridges are introduced simultaneously, the mechanism is not measurement. Ants do not compare lengths. It is differential reinforcement. Ants traveling the shorter bridge complete their round trip faster, return to the nest sooner, and lay a fresh pheromone deposit before the trail has time to decay. Over many cycles, the shorter path accumulates a stronger chemical signal, attracting more ants, producing more reinforcement, compounding the advantage until the shorter route dominates and the longer one is effectively abandoned. This process was first systematically documented in the 1980s and has since been replicated across dozens of species and experimental configurations. What makes it worth attention beyond the behavioral curiosity is the implication: a colony finds the optimal path not by knowing what optimal means, but by running a physical simulation of efficiency using its own bodies as the computational medium. Each ant is simultaneously a data point and a processor. The trail is both the question and the answer, written in molecules that evaporate at a known rate, which means the system even has a built-in mechanism for forgetting paths that stop being useful. The Argentine ant Linipothema humile, native to South America, has spread across 6 continents since the late 19th century. In its introduced range, it has done something that has no precedent among social insects at this scale, formed supercolonies, networks of interconnected nests spanning thousands of kilometers, within which any worker ant can move freely without being attacked by workers from neighboring nests. The largest known supercolony stretches along the Mediterranean coast for approximately 6,000 kilometers from the Italian Riviera to the Atlantic coast of Spain. A second supercolony covers much of coastal California. A third occupies portions of the Japanese coastline. When researchers collected workers from the Mediterranean and Californian supercolonies, colonies separated by an ocean with no contact for over a century, and placed them together, the workers did not fight. They groomed each other. Somehow chemical identity had remained similar enough across a century of geographic separation that nestmate recognition still held. Scientists still debate exactly how this identity is maintained across such distances and timescales, and whether the term supercolony describes a single entity or simply a collection of mutually tolerant populations. What is not debated is the scale. A single cooperative ant network, spanning a continent, is by any measure one of the largest unified animal societies on Earth. Desert ants of the genus Cataglyphis forage alone across open, featureless terrain where there are no landmarks, no pheromone trails left by nestmates, and no visual guides of any kind. They wander in irregular paths, zigzagging, looping, covering distances that can reach 100 meters from the nest entrance. Then, having found food, they turn and walk home in a nearly straight line. This ability is called path integration, or dead reckoning, the same navigational method used by sailors before GPS, who tracked their speed, direction, and time elapsed to calculate position without external reference. The ant maintains a continuously updated internal vector, a running calculation of direction and distance traveled, and at any moment can invert that vector to generate a direct homeward course. Researchers confirmed this by moving ants mid-journey to unfamiliar release sites and watching them walk a path equal in length and direction to their calculated home vector, stopping at the point where the nest entrance should have been, then switching to search behavior when it was not there. The calculation was correct. The nest had simply been moved. What runs this calculation is a brain smaller than a grain of sand, with no access to clocks, compasses, or instruments of any kind. The ant carries its navigation system entirely within itself, updating it continuously step by step for every meter of an unmarked desert. The sky is not uniform. Sunlight, as it scatters through the atmosphere, becomes polarized in patterns that radiate outward from the sun's position in predictable arcs. To a human eye, this polarization is invisible. To a desert ant, it is a compass. Specialized photoreceptors in the dorsal rim of the ant's compound eye, a narrow strip of ommatidia, oriented differently from the rest, are tuned specifically to detect the angle of polarized light rather than its intensity, color. By sampling this angle from multiple points in the sky, the ant can calculate the sun's position even when the sun itself is blocked by an obstacle or cloud cover. This was confirmed in a series of experiments in which researchers placed polarizing filters above outdoor ant foraging arenas and rotated them artificially, shifting the apparent polarization angle. The ants rotated their homeward paths by precisely the same degree. When the filter was removed, they corrected back to the original heading. The system is remarkably robust. It continues functioning under up to 90% cloud cover because polarized light penetrates cloud layers that block direct sunlight. The ant's navigational toolkit is therefore not dependent on clear skies. It reads a signal in the light that most eyes cannot detect from a source that most navigation systems would consider obscured, and produces directional information accurate enough to cross a featureless desert and arrive within centimeters of a nest entrance the width of a pencil. For many years, the dominant assumption was that Cataglyphis desert ants navigated by integrating optic flow, the rate at which the visual field moves past the eye, which correlates with speed and therefore distance traveled. Then a research team tested an alternative hypothesis that the ants were counting steps. They fitted one group of ants with stilts, small bristle extensions glued to their legs, increasing stride length, and shortened the legs of another group by trimming them. Ants with stilts, released at the food source and allowed to walk home, overshot the nest entrance by a distance proportional to the stride length increase. Ants with shortened legs stopped short of it by a corresponding amount. The unmodified control group arrived accurately. The conclusion was that the ants were tracking distance by counting stride cycles, maintaining a running tally of steps taken and using that count as a distance estimate. The mechanism by which this count is maintained remains an open question. Some researchers believe it is processed in a brain region analogous to the insect mushroom bodies, structures associated with memory and learning, while others argue the counting may be distributed across proprioceptive feedback from multiple leg joints simultaneously. What the experiment settled is the more interesting question. An ant crossing a desert alone is keeping score. Teaching in the strict behavioral definition requires that an informed individual modify its own behavior in the presence of a naive individual at a cost to itself, in a way that accelerates the naive individual's acquisition of a skill. By that definition, true teaching has been confirmed in very few non-human species. Ants are among them. In the rock ant Temnothorax albipennis, experienced foragers lead naive nestmates to food sources through a process called tandem running. The leader moves slowly, far more slowly than it would travel alone, pausing frequently to allow the follower to catch up and perform antennal contact taps that signal readiness to continue. If the follower falls behind, the leader waits. If the follower catches up quickly, the leader increases pace. The leader's solo travel speed is measurably faster than its tandem speed, confirming the behavioral cost. The follower, after a successful tandem run, can subsequently navigate to the same food source independently and can itself become a leader. The information transfers. The cost is real. The definition is satisfied. Researchers who documented this in 2006 noted that it was the first confirmed case of teaching in a non-human invertebrate, and that it emerged not from any centralized instruction, but from a two-animal interaction, governed entirely by chemical and tactile signals. No ant in this exchange knows it is teaching, and yet the knowledge moves. A foraging trail is not a static structure. It is a dynamic system that continuously monitors its own performance and adjusts. When a pheromone trail leads to a food source that has been depleted and exhausted by previous foraging runs, returning ants make a small but consequential behavioral change. They do not reinforce the trail on the way home. Without inbound reinforcement, the trail pheromone begins to evaporate. Fewer ants follow a fainter trail. Fewer arrivals mean less reinforcement still. The signal degrades, and within a time frame proportional to the trail's original strength, the route is effectively abandoned. No scout sends a signal saying the food is gone. No manager redirects workers to a new location. The system detects failure through the absence of positive feedback rather than the presence of negative feedback, a design principle that engineers working in network optimization have specifically identified as more stable than alarm-based correction systems. In parallel, other scouts continue ranging in search of new sources, and any successful discovery immediately begins the reinforcement cycle anew. The colony does not mourn a failed trail. It simply stops feeding it, and the path dissolves back into unmarked ground. If you have ever watched an ant trail thin out and disappear over the course of an afternoon, you have watched a living algorithm performing error correction in real time. An ant nest is not a hole in the ground. In mature colonies of species like atta leafcutters, the nest is an engineered structure containing hundreds of distinct chambers connected by a branching network of tunnels built to specifications that maintain internal temperature within a narrow range regardless of surface conditions. Fungal garden chambers are positioned at depths where temperature and humidity stay within the precise tolerances required for fungal growth. Brood chambers are arranged in a thermal gradient, allowing workers to move eggs and larvae to the optimal temperature band as conditions shift. Waste chambers, middens, are positioned at the colony's periphery, both for containment and because their microbial activity generates heat that can be used to warm adjacent tunnels in cooler months. Ventilation shafts of different diameters regulate airflow through convection, drawing fresh air inward at lower openings and expelling warm CO2-laden air through upper channels. No individual ant designed this. Excavation behavior is governed by local rules. Remove soil from where CO2 is high, deposit it where airflow carries it away. The architecture that emerges from millions of these local decisions matches in its functional sophistication what a human engineer would produce after extensive modeling. The difference is that the ant colony builds it without models, without plans, and without knowing what it is building until it is already built. The parallel between ant nest ventilation and human engineering is close enough that it has moved beyond metaphor into direct application. Researchers studying the mound architecture of African termites, whose ventilation systems operate on the same convective principles as ant nests, found that the mounds maintain interior .2 levels and temperature within tight tolerances, despite large fluctuations in external temperature. The mechanism relies on the differential thermal mass of different mound sections. Thick outer walls heat slowly during the day and release that heat slowly at night, driving a convective cycle that continuously cycles air through the structure without any mechanical assistance. Architects in Zimbabwe applied these principles in designing the Eastgate Center in Harare, a building that maintains comfortable interior temperatures without conventional air conditioning, by using passive convective airflow modeled on termite mound architecture. The building uses roughly 10% of the energy a conventional air-conditioned structure of the same size would consume. Ant nests operate on closely related principles, and myrmecologists have noted that the engineering problems the two systems solve are functionally identical. One system was designed by human engineers over several years of study. The other was refined by evolution over 50 million years of trial and elimination. Both arrived at the same solution. The ants got there first. Inside a mature ant colony, waste does not accumulate randomly. Most ant species maintain designated disposal sites, middens positioned with apparent deliberateness at the colony's periphery, away from brood chambers and food stores. Workers carry dead nestmates, exhausted food material, shed larval skins and other organic waste to these zones and deposit them in consistent locations. This behavior is not simply tidiness, it is epidemiology. In a dense colony, unmanaged organic waste is a vector for fungal and bacterial pathogens that can spread rapidly through a population with no immune barrier between individuals. The midden functions as a containment zone, a place where decomposition happens at a controlled distance from the colony's most vulnerable members. Studies of Lasius niger colonies found that waste deposited in middens contained significantly higher concentrations of pathogenic fungi than waste removed before it reached the midden, suggesting that the midden environment itself may function as a managed decomposition zone where certain microbial communities are tolerated or even cultivated to outcompete more dangerous pathogens. Some researchers believe this represents a primitive form of sanitation engineering. Others are more cautious, noting that the behavior could be selected simply for distance from the brood rather than any active management of microbial ecology. The debate continues. The middens, meanwhile, have been doing their job for millions of years, regardless of how humans choose to classify them. In 2018, researchers at the University of Helsinki published findings that wood ants Formica paralugubris deliberately incorporate hydrogen peroxide-producing compounds into their nest material. Specifically, they collect resin from conifer trees and mix it into the nest structure at concentrations that measurably reduce the growth of pathogenic fungi and bacteria within the colony. When colonies were experimentally infected with the fungus, Metahismium-infected colonies increased their resin collection relative to uninfected controls. The behavior change was specific to infection status, not a general increase in foraging activity. A separate body of research has documented ants consuming reactive oxygen species compounds that function similarly to hydrogen peroxide when infected with certain pathogens at dosages that would be mildly toxic to healthy individuals but appear to confer survival advantages to infected ones. The infected ants, in other words, are not simply eating what is available. They are consuming something specifically because it interferes with what is killing them at doses calibrated to the trade-off between toxicity and therapeutic effect. Whether this constitutes self-medication in a meaningful sense, or whether it is a fixed behavioral response that happens to produce medicinal outcomes, is a question scientists still debate with considerable energy. What the experiments confirm is the outcome. Infected ants live longer when they have access to these compounds than when they do not. In 2017, a research team studying Megaponera annalis, the Matabele ant, a specialist predator of termites native to sub-Saharan Africa, documented a behavior with no precise parallel in the social insect literature. During termite raids, Matabele ants sustain significant injuries. Termite soldiers bite and hold onto attacking ants, often severing limbs or damaging leg joints before being dislodged. After a raid, injured workers release a distress pheromone. Uninjured nestmates respond by carrying the injured back to the nest, a rescue behavior documented in other species. What made the Matabele observation distinct was what happened next. Inside the nest, uninjured workers spent between 20 and 60 minutes intensively grooming injured nestmates, specifically targeting the wound sites, licking them repeatedly with apparent focus on the injured area. Ants that received this treatment showed a survival rate of approximately 90%. Ants with equivalent injuries that were isolated and denied treatment died at roughly the same rate. Chemical analysis of the grooming secretions from the metapleural gland and oral gland found antimicrobial compounds at concentrations sufficient to suppress wound infection. The colony was not simply retrieving its injured, it was treating them. Researchers still debate whether the grooming workers can assess injury severity and adjust treatment duration accordingly, or whether they simply respond to pheromone concentration, which itself correlates with injury severity. Among the behaviors that make myrmecologists uncomfortable, assigning simple moral neutrality to ant societies, slave making, ranks near the top of the list. Several ant species, among them Polyergus, Temnothorax, Americanus, and Formica sanguinea, sustain their colonies not by raising their own workers, but by raiding the nests of other species, stealing pupae, and allowing them to eclose hatch inside the raiding colony's nest. The emerged workers, having never known a different colony, perform their behavioral repertoire, foraging, brood care, nest maintenance in service of the colony they hatched into. In the most extreme cases, such as Polyergus, the raiding species has lost the ability to perform basic colony functions entirely. Their mandibles are shaped for combat, not for digging or carrying brood. They cannot feed themselves. Without a continuous supply of enslaved workers, a Polyergus colony starves. Historians and scientists still debate whether the term slavery is appropriate, given the behavioral and cognitive differences between ants and humans, and some researchers argue the term imports ethical frameworks that distort rather than illuminate the biology. Others maintain that the functional description is accurate regardless of inner experience. What is not disputed is the mechanism, one species building its survival on the captured labor of another, generation after generation, with no apparent diminishing of either colony's participation in the arrangement. Approximately 50 million years ago, roughly 25 million years before the earliest evidence of human agriculture, a lineage of ants in South America began cultivating fungi. The leafcutter ants of the tribe Attini are the most visible descendants of that innovation, but the agricultural system they represent is far older and more complex than the common image of ants carrying leaf fragments suggests. The leaves are not food. They are substrate, raw material cut and carried to underground garden chambers, where they are chewed, processed with ant fecal droplets containing specific enzymes, and used to cultivate a fungus of the genus Leucoagaricus that the ants then harvest as their primary food source. This fungus no longer exists in wild form. It has been cultivated in continuous ant colonies for so long that it has lost the ability to reproduce independently and bears domestication markers comparable to those seen in human-cultivated crop plants. The ants, in turn, are dependent on the fungus. They cannot digest the leaves directly. The relationship is one of mutual obligate dependence that has persisted across 50 million years of environmental change, continent drift, and mass extinction events. It is by any reasonable measure the longest-running agricultural enterprise in the history of life on Earth. The first human farmers appeared roughly 10,000 years ago. The ants had already been farming for 49,990,000 years before that. Most ant species possess a structure called the metapleural gland, a paired gland located near the junction of the thorax and abdomen, with no equivalent in any other insect order. It secretes a complex mixture of compounds, including phenylacetic acid, indole, acetic acid, and various short-chain fatty acids, many of which have documented antimicrobial and antifungal activity. In a colony where thousands to millions of individuals live in close contact in warm, humid underground chambers, conditions that would ordinarily constitute an ideal environment for epidemic disease, the metapleural gland secretion functions as a colony-wide prophylactic worker's spread the secretion through grooming, contaminating the nest substrate and each other's cuticles with a continuous low-level antimicrobial coating. Pharmaceutical researchers have taken notice. Several compounds isolated from ant metapleural gland secretions have shown activity against antibiotic-resistant bacterial strains in laboratory conditions, including some strains of Staphylococcus. The compounds are not yet in clinical use, and the path from laboratory activity to clinical antibiotic is long and uncertain. But the underlying observation that ants have been managing epidemic disease in dense populations for millions of years using chemical tools that human medicine has not yet fully catalogued has made the metapleural gland one of the more actively studied structures in applied entomology. The colony's pharmacy has been open longer than ours. We are only beginning to read its inventory. When a rock ant colony of the species Temnothorax must choose between two potential nest sites, one superior, one inferior, the decision process unfolds through a mechanism called quorum sensing. Individual scouts explore candidate sites independently and assess their quality using a set of criteria: entrance, size, interior volume, darkness, dryness, and distance from the current nest. A scout that finds a high-quality site begins recruiting nestmates to visit it, but does not commit the colony to moving until it has encountered a threshold number of other ants already present at that site. Below the quorum threshold, the scout recruits cautiously. Above it, recruitment accelerates sharply, and the colony begins transporting brood and nestmates to the new location. This quorum threshold serves a critical function. It prevents the colony from committing to a site based on a single scout's assessment, while ensuring that decisions are still made quickly enough to be useful when urgency demands it. When colony size is experimentally reduced, removing workers until the colony is small, the quorum threshold adjusts downward, making the colony more decisive but less accurate. Larger colonies maintain higher thresholds and make better average decisions at the cost of speed. The colony, in other words, has a decision-making architecture that scales its accuracy with the stakes, and it achieves this without any individual ant ever knowing what a quorum is. Individual ant memory is modest by vertebrate standards. Most species show reliable retention of spatial and chemical information over periods of hours to days, with some documented cases extending to weeks in species with longer-lived workers. What makes ant memory ecologically interesting is less its duration than its architecture. Individual memory and colony-level information retention are separate systems operating in parallel. An individual Cataglyphis forager can remember the location of a productive food site across multiple foraging bouts spanning several days, using a combination of path integration memory and landmark recognition. At the colony level, information persists far longer than any individual's memory, because the pheromone trail encoding a productive route can be maintained continuously by successive generations of foragers who have never independently discovered the route, each reinforcing a signal laid by predecessors they never met. A trail first established by ants that are now dead can remain active and accurately followed years later, maintained by the accumulated deposits of workers who simply followed it and added to it. The colony remembers through its infrastructure routes that no living ant discovered. Think of it as the difference between a person's recollection and a city's roadmap. The roads outlast everyone who built them, and each driver who uses them reinforces their existence without knowing their origin. A colony is not a frictionless cooperative. Inside even a well-functioning ant society, individual workers sometimes pursue reproductive interests that conflict directly with the colony's collective strategy. In species where workers retain functional ovaries, as in many Diacama and Camponotus species, some workers will attempt to lay unfertilized male eggs during periods when the queen's reproductive suppression weakens. These workers do not announce their activity. They lay eggs surreptitiously, often in peripheral nest areas away from the queen's chemical influence, and in some documented cases, disguise the eggs chemically to reduce their detectability by policing workers. The colony has a counter-response worker policing, in which other workers actively seek out and destroy eggs not laid by the queen using chemical cues to distinguish queen-laid eggs from worker-laid ones. The accuracy of this policing varies by species and colony condition. In species where workers are closely related to the queen's offspring, policing is more stringent, the inclusive fitness logic is tighter. In species where relatedness is lower, some worker reproduction is tolerated. Researchers studying this dynamic have described it as an arms race between reproductive deception and detection, playing out at a chemical level inside the colony. Invisible to any outside observer, the colony appears unified. Beneath that surface, the same tensions between individual and collective interest that characterize human institutions operate continuously in the dark in the language of molecules. When an ant dies inside a nest, the body does not remain where it falls for long. Within a day or two, sometimes within hours, workers remove it to the midden. The trigger for this behavior is chemical. As an ant's body begins to decompose, it releases a specific blend of fatty acids, primarily oleic acid, that serves as a death signal. Workers detecting this blend pick up the source and carry it to the disposal zone regardless of what the source is. Researchers demonstrated this with a now classic experiment. They applied oleic acid to live healthy ant workers and placed them back in the colony. The treated ants were immediately picked up by nestmates and carried to the midden alive and walking. They were returned to the nest. They were carried to the midden again. This continued until they groomed the chemical signal off their own bodies, at which point nestmates stopped removing them. The colony does not assess whether a nestmate is alive or dead through observation of movement or behavior. It reads a chemical signature and acts on the signature alone. An ant covered in the smell of death is to its colony dead, regardless of evidence to the contrary. The system is efficient and largely reliable because the circumstances under which a healthy ant acquires a death chemical signal in nature are essentially zero. The experiment worked because it was, from the colony's perspective, an impossible situation. Laboratory studies of ant problem-solving have produced results that continue to generate debate about where the boundary lies between instinct and cognition. In one widely replicated experimental design, ant colonies are presented with a U-shaped barrier between the nest and a food source, a configuration that requires the forager to move away from the food before finding a path around the barrier. Naive foragers initially persist in direct approaches, repeatedly contacting the barrier. Over multiple trials, successful routes are found pheromone reinforced, and the colony converges on the solution. What varies between species and what researchers find most interesting is the rate of convergence and whether individual ants show any evidence of latent learning, retaining spatial information gathered during unsuccessful trials that accelerates the discovery of the correct route on subsequent attempts. Some species show measurable latent learning effects. Others appear to rely entirely on pheromone reinforcement, with no individual retention of failed path information. In bridge-building experiments where ants must span a gap of variable width, some species assemble multi-ant chains of precisely calibrated length, stopping when the bridge is long enough and no longer. Researchers still debate whether this calibration involves any form of assessment by individual ants, or whether it is entirely an emergent property of the group dynamics involved in chain formation. The behavioral algorithms that govern ant foraging lay pheromone on successful paths, allow unsuccessful ones to decay, follow concentration gradients, adjust output based on traffic, have been formalized into a family of computational methods called ant colony optimization, first developed by the computer scientist Marco Dorigo in the early 1990s. These algorithms have since been applied to problems in logistics, routing, telecommunications, network management, protein folding, prediction scheduling, optimization, and robotic swarm coordination. The traveling salesman problem, finding the shortest route connecting a set of points, is one of the most computationally demanding problems in combinatorics. And ant colony optimization consistently produces near-optimal solutions faster than many classical approaches on large-scale instances. UPS and other logistics companies have used variants of these algorithms to optimize delivery routing across thousands of vehicles and stops. NASA has explored ant-inspired coordination protocols for autonomous rover swarms. The underlying principle in every application is the same one operating in an ant trail: no central controller, no global knowledge, no explicit optimization, just local rules applied by many agents simultaneously producing global efficiency. As an emergent property, the ant colony was not trying to solve the traveling salesman problem, it was trying to find food. Are structurally identical. The caste system of an ant colony is not entirely fixed. In most species, the developmental pathway worker, soldier, queen is determined during the larval stage and cannot be reversed. In a small number of species, however, adult workers retain the physiological capacity to become functional reproductives under specific conditions. In Harpegnathos saltata, the Indian jumping ant, workers can transition to a gamergate state, a reproductively active worker, when the queen dies. Competing workers engage in ritualized antenna dueling tournaments, and the winners begin producing eggs within weeks. Their brains change measurably. The mushroom body's neural structures associated with learning and memory shrink, while regions associated with reproductive physiology expand. The transition is accompanied by a significant lifespan extension. Gamergates live 5 times longer than workers approaching queen-level longevity. Crucially, this transition can be reversed if a gamergate is reintroduced to a colony with an active queen. The same individual brain can reorganize itself in either direction depending on social context. Researchers studying this plasticity have identified specific genes, particularly those regulated by juvenile hormone, that appear to act as molecular switches governing the transition. The finding suggests that the gap between worker and queen, at least in some species, is not a wall but a threshold one. The colony can push individuals across when circumstances require it. Worker ants do not sleep the way most people imagine sleep. A long, consolidated period of rest, followed by sustained wakefulness. Instead, they cycle through hundreds of brief rest episodes distributed across the full 24-hour day. Research on fire ant workers found that individuals average 253 sleep episodes per day, each lasting approximately 1.1 minutes, for a total daily sleep time of just under 5 minutes per 24-hour period. During these micro-rests, the ant's antennae drop and cease movement, the most reliable behavioral indicator of sleep state in insects. Queen ants sleep differently: fewer episodes, longer duration, more consolidated rest periods, with total daily sleep time approaching 9 hours. The asymmetry matters for colony function. Because workers cycle through rest at different phases, the colony never has all workers simultaneously inactive. There is always a population of alert individuals available to respond to disturbance. The queen's longer, deeper sleep cycles may reflect the greater physiological demands of continuous reproduction, or they may simply reflect the reduced need for constant environmental vigilance that comes with living in the colony's most protected interior chamber. Some researchers have noted that the brief, fragmented nature of worker sleep resembles the polyphasic sleep patterns documented in other high-vigilance animals, suggesting that the colony, through the distributed sleep schedules of its members, has solved the same problem a hospital night shift solves with rotating personnel. The chemical language of ants is not a simple system of alarms and trail markers. In the fire ant Solenopsis invicta alone, researchers have identified over 20 distinct pheromone compounds, each produced by different glands and carrying different information. The Dufour's gland produces trail pheromones with directional information. The poison gland contributes alarm components. The postpharyngeal gland carries nestmate recognition compounds. The rectal gland secretes compounds used in territorial marking. Each signal operates at a different concentration range, evaporates at a different rate, and is detected by different chemoreceptor subtypes on the antenna. Some signals are binary present or absent, while others carry graded information, where concentration encodes urgency or distance. Alarm pheromones, for instance, are released in quantities proportional to the threat level, and workers in the vicinity respond with recruitment behavior scaled to the concentration they detect. A low concentration triggers alert posture and local investigation. Like a volume dial rather than a simple switch, researchers have documented at least a dozen distinct behavioral responses that can be reliably triggered by specific pheromone blends, suggesting a combinatorial capacity and multiple compounds in varying ratios, producing distinct meanings that may extend the effective vocabulary of the system far beyond the count of individual compounds. The language is chemical. Its complexity is only beginning to be mapped. When an ant colony faces sudden threat, a predator breaching the nest entrance, a competing colony crossing a territorial boundary, an intruder of any size, the response does not begin with visible activity. It begins with a molecule. The alarm pheromone in most species, a volatile compound released from the mandibular gland, diffuses outward from the source ant in an expanding sphere that decreases in concentration with distance. Ants within the inner zone of high concentration closest to the threat switch immediately to aggressive or defensive behavior. Mandibles, open bodies orient toward the pheromone source movement accelerates. Ants in the outer lower concentration zone shift into an alert state. Increased speed, heightened antennal activity, readiness to respond but not yet committed to a direction. This creates a spatial gradient of response that concentrates defensive action at the point of threat while priming the broader colony for escalation. The alarm signal degrades rapidly. Most alarm pheromones have half-lives measured in seconds to minutes, which means the alert automatically stands down if the threat is resolved or the source ant stops releasing the signal. There is no all-clear message required. The silence is the all-clear. This auto-cancelling feature prevents false alarms from propagating indefinitely through the colony, a design principle that, as any systems engineer will recognize, is considerably more elegant than the human equivalent. Trophylaxis is the mouth-to-mouth transfer of liquid food between colony members, and in most ant species it is among the most frequent social interactions occurring inside the nest. A forager returning with a crop full of liquid food, nectar, plant sap or dissolved nutrients from prey does not simply deposit the load in a storage chamber. It is met by nestmates who solicit food by tapping the forager's mouthparts with their antennae, triggering regurgitation of a droplet that the soliciting ant consumes. This exchange is not merely nutritional. Research has shown that the liquid transferred during trophylaxis contains a complex mixture of compounds beyond food, hormones, proteins, and chemical identity markers that circulate through the colony via this network of sharing. Queen-produced compounds that suppress worker reproduction travel through trophylaxis to reach workers throughout a large colony. Immune components, compounds that may prime nestmates' immune responses, have also been documented in trophylactic fluid. A colony connected by active trophylaxis is not simply sharing calories. It is sharing a biological medium that carries chemical information, reproductive signals, and potentially immune priming across thousands of individuals simultaneously. Some researchers have described a mature ant colony's trophylactic network as functionally analogous to a circulatory system, one that distributes not blood, but a colony-wide chemical conversation continuously updated, moving through every individual in the nest. The term superorganism was coined by the entomologist William Morton Wheeler in 1911, and it has been debated, refined, and contested ever since. At its core, the concept proposes that an ant colony functions as a single biological entity, that the appropriate unit of analysis is not the individual ant but the colony as a whole, in the same way that the appropriate unit of analysis for a human body is not an individual cell but the organism those cells compose. The analogy holds with unexpected precision in several directions. Workers performing different tasks correspond to differentiated cell types. The queen's reproductive function corresponds to germ cells. The trophylactic network corresponds to a circulatory system. Pheromone gradients function as hormonal signals. Immune responses to pathogens are coordinated at the colony level rather than the individual level. The colony grows, reaches maturity, reproduces by sending out new queens, and eventually senesces and dies, a life cycle that maps onto organismal biology more cleanly than most metaphors do. Critics of the superorganism concept argue that it obscures the genuine conflicts of interest between individuals within a colony, the cheetah workers, the competing queens, the caste tensions documented in many species. Proponents respond that similar conflicts exist between cells in a body, cancer being the most obvious example, without invalidating the organismal framework. The debate has not been resolved. The colony, meanwhile, continues functioning as though the question does not concern it. The question of whether ants form something resembling social preferences, consistent individual-specific associations that go beyond functional task coordination, was largely ignored until researchers began tracking individual ants within colonies using paint marks and, more recently, radio frequency identification tags. What emerged from these studies was unexpected. In several species, including Camponotus, carpenter ants, and Formica wood ants, individual workers show statistically non-random association patterns. Certain pairs of workers are found together, groom each other, and share food more frequently than chance would predict across multiple observation sessions spanning days or weeks. These associations are not fully explained by task overlap, age cohort, or spatial proximity within the nest. Some researchers have characterized these patterns as rudimentary social bonds, consistent preferential associations that influence behavior in ways analogous at a vastly simpler level to social relationships in vertebrates. Others argue the term bond imports too much cognitive weight, and that the patterns are better explained by chemical similarity workers whose cuticular hydrocarbon profiles are most similar, gravitating toward each other during grooming as a byproduct of nestmate recognition chemistry. The mechanism remains contested. What the data consistently shows is that inside a colony of thousands, some pairs spend measurably more time together than others, and that this is not random. In a colony of hundreds of thousands of workers, the queen is never formally introduced. There is no ceremony, no announcement, no visual display of status, and yet workers throughout the colony behave differently in her presence than in her absence, grooming her with specific attentiveness, feeding her preferentially, orienting toward her, and in many species, forming a retinue of workers that follows her movements through the nest. The mechanism is chemical. Queens produce a blend of compounds collectively called queen pheromone or queen signal from multiple glands, including the Dufour's gland, the poison gland, and cuticular secretions that vary in composition by species. These compounds travel through the colony via trophylaxis and direct contact, suppressing the reproductive physiology of workers and simultaneously triggering their attentive behavior toward the queen. Workers that detect high queen signal concentrations reduce their own ovarian activity and increase behaviors associated with queen care. As queen signal concentrations drop because the queen is aging, because colony size has grown too large for the signal to distribute evenly, or because the queen has died, workers' physiology begins to shift. Ovaries activate, behavioral repertoires change. The colony begins the process of replacing or supplementing its reproductive center. The queen's authority in this sense is entirely pharmacological. She does not rule by presence, she rules by chemistry. Queens are not permanent. In several ant species, workers execute their own queen under specific circumstances. A behavior called regicide, documented with sufficient regularity, that it is now considered a standard feature of colony life in certain lineages rather than an aberration. The triggers vary by species. In Solenopsis invicta fire ants, the presence of a gene variant called the social chromosome determines whether a colony tolerates one queen or multiple queens. Workers carrying one variant kill every queen except one. Workers carrying the other variant tolerate polygyne colonies with many queens simultaneously. When a queen begins to decline in fertility, producing fewer eggs, releasing weaker chemical suppression signals, workers in some species become measurably less attentive to her, and in some documented cases, actively aggressive. The transition from neglect to execution can happen within days of the fertility decline, becoming detectable through chemical signals. In species that practice seasonal reproduction, Established queens may be killed at the end of the reproductive season and replaced by new queens that the colony has been raising in parallel. The colony does not mourn. Workers that participated in the queen's care may participate in her death within the same season, governed not by loyalty or betrayal, but by the same chemical gradients that governed every other decision the colony has made. What looks from the outside like political violence is from the inside a response to a shift in molecular signal. Aphids are small, soft-bodied insects that feed on plant sap, extracting more sugar than their metabolism can use and excreting the excess as a liquid called honeydew. Ants drink honeydew. This exchange, which sounds incidental, has in many species pairs become an elaborate mutualism refined over millions of years into something that resembles in its structural logic the relationship between humans and domesticated livestock. Ant workers milk aphids by stroking their abdomens with their antennae, stimulating honeydew production. In return, workers defend aphid colonies from predators, ladybirds, lacewing larvae, parasitic wasps, with the same aggression they would bring to defending their own brood. Some ant species carry aphid eggs into their nests to overwinter, then transport the hatched aphids back to host plants in spring. Several ant species have been documented removing aphid wings, either by grooming them away or by releasing chemicals that suppress wing development, reducing the aphid's ability to disperse to other plants and keeping them within the ant's foraging range. The aphids in these interactions have effectively lost autonomous mobility and exist under the management of the ant colony. Whether this constitutes domestication in the same sense the word applies to cattle is a question that agricultural historians and entomologists approach differently, and the debate has not reached consensus. The whistling thorn acacia of East Africa, Vachellia drepanolibium, maintains one of the most studied ant-plant relationships on Earth. The tree provides two resources to resident ant colonies, primarily Chromatogaster species that are found almost nowhere else in the savanna, hollow, swollen thorns that serve as pre-built nest chambers, and nectar-secreting organs called extrafloral nectaries that produce food outside of any flower. In return, the ants patrol the tree, continuously attacking any herbivore that touches the branches, from insects to elephants. Researchers have documented elephants retreating from acacia trees with active ant colonies after ants swarmed into their trunks and nasal passages. The trees with active ant colonies suffer measurably less herbivore damage and grow faster than trees where ant colonies have been experimentally removed. The relationship is specific. Different ant species that colonize the same tree provide different levels of protection, and the tree's investment in each species to the amount of nectar produced. The number and size of hollow thorns adjusts accordingly over time. When researchers removed ants from trees and excluded them for several years, the trees responded by increasing the production of chemical defenses, tannins and other compounds that deter browsing, as though switching from a biological security system to a chemical one when the biological option was unavailable. The tree and the ant have been co-evolving this contract for long enough that both parties have developed contingency plans for when the other fails to hold up their end. The large blue butterfly Phengaris arion spends most of its larval life inside red ant nests, not as a captive, but as a welcomed guest fed by the ants, protected by the ants, and prioritized over the ant's own brood during food shortages. It achieves this through chemical mimicry of extraordinary precision. In its early instars, the caterpillar feeds on thyme flowers and emits surface chemicals that cause Myrmica ant workers to behave as though it is a lost larva, picking it up and carrying it into the nest. Once inside, the caterpillar produces acoustic signals, stridulation, that mimic the sounds made by Myrmica, queens triggering worker behaviors associated with queen care, intensive grooming, preferential feeding, and protection. Worker ants fed the caterpillar before feeding their own larvae during food scarcity. The caterpillar grows fat in the nest for approximately 10 months, overwinters as a pupa, and emerges as an adult butterfly that exits the nest before the ants recognize the deception, if they ever do. The butterfly went extinct in Britain in 1979 when changes in land management altered thyme availability and consequently Myrmica ant populations. It was reintroduced successfully in the 1980s after researchers identified the precise Myrmica species required and the habitat conditions that supported them. Saving the butterfly required saving the ant first, which required understanding the chemistry well enough to restore the habitat that sustained both. Ants are not simply members of ecosystems in many environments. They are foundational to how those ecosystems function. A single hectare of Amazon rainforest may contain over 8 million individual ants representing hundreds of species, each occupying a distinct ecological role. Collectively, ants in tropical forests turn more soil than earthworms, move more seeds than any other animal group, and regulate the populations of more prey species than any vertebrate predator in the same habitat. The term ecosystem engineer, applied to animals whose activities physically modify the environment in ways that create habitat for other species, applies to ants with more justification than it applies to most animals it is used to. Describe. Ant tunneling increases soil porosity and water infiltration rates. Ant middens create nutrient hotspots that support distinct plant communities. Ant-guarded plants grow in configurations shaped by the ant's territorial behavior rather than by light availability alone. Remove ants from a tropical forest plot, as researchers have done in controlled exclosure experiments, and the changes cascade through plant composition, soil chemistry, arthropod diversity, and small vertebrate populations within months. The ecosystem does not simply lose a component, it reorganizes around the absence. Most people encounter ants as a peripheral annoyance. The forest encounters them as load-bearing structure. Several ant species do more with their dead than simply remove them to a midden. In Novomessor coccarelli, and certain Pogonomyrmex harvester ants workers have been observed burying dead nestmates, covering bodies with soil particles, nest material, or debris in a deliberate, repeated manner that researchers distinguish from ordinary refuse disposal by its location and the effort invested. The burial sites tend to be within or adjacent to the nest, rather than at peripheral midden zones. Workers return to the site repeatedly, adding material over several hours. Whether this constitutes burial in any meaningful cognitive sense, or whether it is a response to a specific chemical signal that happens to produce burial-like behavior, remains an open question. What adds complexity to the observation is that the same workers who bury nestmates in some contexts carry them to the midden in others, suggesting that the behavior is context-dependent rather than fixed. Some researchers have proposed that the distinction may relate to the cause of death. Ants killed by certain pathogens may release different chemical signals than ants that died of age or injury, triggering different disposal responses calibrated to the infection risk. The colony in this reading does not grieve, it performs risk assessment. The behavior that resembles mourning may be at its core a sophisticated epidemiological response, which does not make it less remarkable, only differently so. Ant territory is not marked with visible boundaries. It is written in molecules renewed continuously and readable only to the animals whose chemistry evolved to read it. Most ant species deposit territorial pheromones from the hindgut or Dufour's gland along the edges of their foraging range, creating a chemical perimeter that nestmates recognize and respect, and that intruders detect as warning. The precision of this system is considerable. Researchers tracking pheromone boundaries between neighboring Lasius colonies found that the chemical frontier was maintained within centimeters of a stable line over periods of months, shifting gradually as colony sizes changed and resources fluctuated. When two colonies' territorial margins meet, the boundary zone becomes a site of continuous, low-level conflict, frequent encounters between workers that escalate to fighting or retreat, depending on relative chemical signal strength and worker density. The colony with more workers in the boundary zone producing more pheromone tends to push the line outward. The colony with fewer workers retreats it. Territory in this system is not a fixed property, but a dynamic equilibrium maintained by the continuous chemical output of living bodies. When a colony shrinks through disease, resource shortage, or queen loss, its territorial margin contracts automatically because fewer workers produce less signal and the balance tips toward the neighbor. No negotiation occurs. No treaty is signed. The boundary moves because the chemistry changes. The queen does not simply exist at the center of the colony. She broadcasts through a continuous output of pheromonal compounds distributed via Trophylaxis, direct contact, and volatile diffusion through the nest atmosphere. The queen maintains a chemical field that shapes the physiology and behavior of every worker in the colony simultaneously. This field carries multiple messages layered into a single chemical blend. One component suppresses worker ovarian development, the reproductive suppression signal. Another triggers worker attentiveness and grooming behavior toward the queen. A third may signal the queen's identity and health status, allowing workers to assess whether replacement should be initiated. Researchers studying Lasius niger found that workers at different distances from the queen received different concentrations of these signals, creating a gradient within the colony. Workers nearest the queen showed the strongest reproductive suppression and the most intense queen care behavior. Workers at the colony's periphery receiving diluted signal showed measurably higher ovarian activation and less queen-directed behavior. The queen's influence, in other words, is not uniform. It attenuates with distance, the way a radio signal weakens as you move away from the transmitter. Large colonies where the dilution effect is most pronounced show higher rates of worker reproduction at their margins. The queen rules most completely in the space closest to her, and her authority becomes genuinely approximate at the edges of a colony she may never physically visit. Ants are not silent. Most species produce sound through stridulation, a process in which a file-like ridge on one body segment is scraped across a corresponding scraper on an adjacent segment, generating vibrations that travel through the substrate rather than through air. The sounds produced are not audible to human ears under normal circumstances, but they are detectable with sensitive microphones pressed against the nest surface, and they carry specific information. In Myrmica ants, stridulation has been documented in the context of distress workers trapped under debris or soil collapse, stridulate in patterns that elicit digging behavior from nestmates on the other side of the blockage. Rescue operations, coordinated by acoustic signals, have been filmed in laboratory conditions, with workers orienting their excavation toward the stridulation source and adjusting direction as the signal shifts. Queens of several species produce distinct stridulation patterns that differ from worker patterns in frequency and temporal structure, and workers respond to queen stridulation with behavioral deference not triggered by worker-produced sounds. In the leafcutter genus Atta, researchers have found that workers stridulate during the cutting process, and that the vibrations generated travel down the leaf fragment into the cutting surface, possibly functioning as a quality assessment tool, with feedback from the vibration pattern informing the ant about the leaf's toughness and suitability for fungal cultivation. The ant is, among its other accomplishments, a musician, a very quiet one, playing for an audience that listens through its feet. The systematic rescue of trapped nestmates has been documented in multiple ant species, but the Matabele ant Megaponera analis, already noted for its wound treatment behavior, provides the most thoroughly studied example of coordinated rescue under field conditions. During termite raids, individual Matabele workers are frequently seized by termite soldiers and held immobile, mandibles locked onto leg or body segments. These captured ants release a distress pheromone that draws free nestmates to their location. The responding ants do not simply attack the termite holding the captive. They engage in a targeted retrieval, gripping the captured ant and pulling it free, sometimes requiring sustained effort, lasting several minutes against a termite that is itself anchored and resisting. In experiments where researchers artificially trapped ants using small wire snares, nestmates responded to the distress signal by approaching and working to free the trapped individual, abandoning the effort only after extended failure. The evolutionary logic of rescue is straightforward in a species where individual workers are highly trained specialists. A Matabele raiding ant that has participated in multiple termite raids carries accumulated tactical experience that the colony cannot instantly replace. Rescuing experienced individuals preserves that embodied knowledge, Researchers calculated that active rescue behavior increases post-raid colony survival rates measurably, and that colonies denied the ability to rescue their injured declined significantly faster than colonies where rescue was permitted. When a colony loses its queen to age, predation, or disease, it faces an existential problem. Without a reproductive center, the colony cannot replace its workers, and an aging worker population will decline to extinction within months. The colony's response to this crisis varies by species, but in several it includes a behavior that at first appears socially improbable: the adoption of a foreign queen. In Formica, wood ants and several lazier species orphaned workers will accept a mated queen of the same or closely related species who enters the nest, even one that has never had contact with these workers before. The acceptance process is not immediate. The foreign queen is initially treated with suspicion, subjected to intense inspection, and sometimes attacked. Over a period of hours to days, if the queen's chemical profile is sufficiently compatible with the colony's recognition template, workers shift from aggression to tolerance, then to active care. The queen begins laying eggs. The colony continues. Researchers studying this process have found that the threshold for acceptance shifts dynamically with the colony's desperation. Orphaned colonies with few remaining workers accept foreign queens at higher rates and with less chemical scrutiny than orphaned colonies that still have substantial worker populations. The colony in its most acute need lowers its standards, a behavioral flexibility that has likely saved lineages that would otherwise have terminated when their original queen was lost. Ant wars are not permanent states. They have beginnings, escalations, and ends, and the mechanisms that terminate them are as structured as the ones that initiate them. In Formica wood ant territorial conflicts, the transition from active combat to truce follows a pattern that researchers have described as negotiated through chemical and behavioral signaling rather than through exhaustion alone. As one colony gains advantage, pushing its territorial margin forward, increasing worker density in the contested zone, the losing colony's workers shift from offensive to defensive postures, and the rate of combat initiations from the work of, say, losing side decreases. The winning colony, encountering less resistance, reduces its own patrol intensity in the contested area. The boundary stabilizes at a new position. In species that engage in tournament behavior, ritualized displays of colony strength that substitute for direct combat, the resolution is more explicit. Myrmica and certain Formica species conduct mass displays at territorial boundaries, with workers from each colony forming dense aggregations that face each other, assess relative size, and then disperse without fighting. The larger aggregation typically prevails, and the smaller colony retreats its boundary. The assessment appears to be accurate. Colonies rarely escalate to direct combat when the size disparity is large, and escalation is more likely when aggregations are closely matched. Scientists still debate whether individual ants are capable of assessing opponent numbers directly, or whether the assessment emerges from collective response dynamics that no individual ant performs alone. The Argentine ant supercolony presents one of the most puzzling identity recognition problems in animal behavior. Workers separated by thousands of kilometers, with no possibility of direct contact between individuals belonging to populations that have been genetically isolated for generations, nonetheless recognize each other as nestmates when brought together in laboratory conditions, grooming rather than fighting, tolerating shared space, treating each other as colony members despite having never shared a colony. The mechanism appears to involve cuticular hydrocarbons, waxy compounds on the ant's surface that serve as chemical identity badges. Within the supercolony, these profiles are sufficiently similar across populations that workers' recognition systems classify them as nestmates. Rival supercolonies, the Mediterranean supercolony and a smaller supercolony also present in southern Europe, have divergent hydrocarbon profiles, and workers from these two groups fight immediately when introduced. The divergence between fighting and non-fighting populations does not correspond to genetic distance in any simple way. Some genetically distant populations share hydrocarbon profiles and tolerate each other. Some genetically close populations have diverged in profile and fight. The chemical identity badge, not the genetic relationship, determines the social response. This finding has implications that extend beyond ants. It suggests that in sufficiently large social systems, shared identity can be maintained through a chemical medium across distances and timescales that preclude any direct social connection between the individuals who share it. A colony founded by a single queen, as most ant colonies are, follows a developmental arc that unfolds over years or decades and mirrors in structural outline the life cycle of an organism. The founding stage begins when the mated queen seals herself into a small chamber and raises her first workers entirely on the metabolized energy of her own flight muscles and fat reserves, producing a cohort of small undernourished workers called nanitics. These founding workers are the colony's infrastructure investment, smaller and shorter lived than later workers, but sufficient to begin foraging and expanding the nest. As resources increase, worker size and specialization increase with them, and the colony enters a growth phase that may last several years. Mature colonies in most species reach a characteristic size ceiling determined by resource availability and species-specific parameters, then stabilize. Reproduction, the production of new queens and males for nuptial flights, typically begins only after the colony has reached a threshold size, often years after founding. The colony's final stage, senescence, begins when the queen's fertility declines. Worker production slows. The population ages. The colony's behavior becomes less responsive, its foraging range contracts, and over months or years it diminishes to zero. Nothing dramatic marks the end. The last workers simply stop being replaced. The tunnels remain. The chemistry fades. And somewhere nearby, in a crack in the soil, a newly mated queen is sealing herself into a small chamber alone, beginning again. The oldest confirmed ant fossil is a worker of the species Spheca freyi, preserved in amber from what is now New Jersey, dated to approximately 92 million years ago. It is recognizably an ant, 6 legs, a petiole, differentiated body segments, and yet it carries features that mark it as transitional, long, straight mandibles, unlike the curved or toothed mandibles of modern ants, and a body plan that sits visibly between the wasp ancestor and the ant descendant. Amber preservation captures detail that stone fossilization cannot. The Spheca specimen retains surface texture, leg joint articulation, and antennal structure clear enough to allow meaningful comparison with living species. Since its discovery in 1967, dozens of additional Cretaceous ant species have been described from amber deposits in Burma, France, Canada, and the Baltic region, filling in portions of the ant's early evolutionary history with a resolution unusual in paleontology. What the fossil record shows collectively is that ants were rare during the Cretaceous present but not dominant, representing a small fraction of insect diversity in amber samples from that period. Then, approximately 60 million years ago, their representation in the fossil record increases sharply and dramatically. Something changed. The leading hypothesis connects this expansion to the concurrent rise of flowering plants, a world being remade by angiosperms, offering new ecological opportunities that ants with their generalist social architecture were positioned to exploit. The forest changed. The ant was ready. Ants are wasps. Not metaphorically, not ancestrally in a distant or approximate sense, but in the direct and specific way that a daughter is her mother's child. The ant lineage descends from within the wasp family. Vespoidea, most likely from a group of ground-hunting wasps, similar to modern Typhoidae. Sometime in the mid-Cretaceous period between 140 and 168 million years ago. The evidence comes from both morphology and molecular phylogenetics, the analysis of genetic sequences to reconstruct evolutionary relationships. Several features that distinguish ants from other insects, the petiole, the metapleural gland, the elbow-jointed antenna, are modifications of structures already present in wasp ancestors, refined over millions of years of subterranean life. The transition to sociality, the development of sterile worker castes dedicated to colony maintenance rather than personal reproduction, is the defining evolutionary step, and it appears to have occurred once in the ant lineage, producing a single common ancestor from which all approximately 20,000 known ant species descend. The social architecture that seems, from a human perspective, to be ants' most remarkable feature, was in place before ants were recognizably ants, before the petiole took its modern form, before the mandibles diversified, before the first colony expanded beyond a handful of workers. Sociality was not a late refinement. It was the foundation on which everything else was built, and on which 100 million years later, everything else still rests. The explosive diversification of ants in the fossil record coincides with a precision that is difficult to dismiss as coincidence. With the explosive diversification of flowering plants, the angiosperms, during the late Cretaceous and early Paleogene, the two radiations were not independent. Ants dispersed seeds, protected plants from herbivores, and aerated the soil around root systems. Plants produced extrafloral nectaries that fed ant colonies, hollow structures that housed them, and chemical signals that recruited them as defenders. Each lineage created ecological opportunity for the other, and the expansion of each drove the expansion of the other in a feedback loop that reshaped terrestrial ecosystems across the entire planet. The forests that exist today, the Amazon, the Congo Basin, the forests of Southeast Asia, are, in a meaningful sense, the product of this coevolution. Their structure, their species composition, the way their canopy is organized and their soil is built, all bear the imprint of 100 million years of mutual shaping between ants and the plants they live among. Historians of evolution sometimes describe this period as one of the great transformations in the history of life on land. The emergence of a world organized around flowers, fruit, and the animals that moved them. The ant was not a passenger in that transformation. It was one of its primary architects, working without intention, driven entirely by the logic of survival and producing. Across geological time, a world that neither it nor the plants it partnered with could have built alone. 66 million years ago, the Chicxulub impactor struck what is now the Yucatan Peninsula and triggered a mass extinction that eliminated approximately 3/4 of all species on Earth, including every non-avian dinosaur. The ants survived. They were not unaffected. Ant diversity almost certainly contracted during the impact winter that followed when sunlight was blocked. Temperatures dropped, and the plant communities that supported ant food webs were devastated globally. The features that allowed survival were probably the same features that had always made ants ecologically resilient: subterranean nesting that provided insulation from surface temperature extremes, dietary flexibility that allowed colonies to shift between food sources as availability changed, and a colonial structure in which the loss of individual workers did not terminate the colony's capacity to function. Smaller body size also correlates with survival across multiple mass extinction events. Smaller animals require less food and can maintain populations on resources insufficient to support larger ones. The ant is small. Its nest is underground. Its diet is flexible. Its colony can absorb enormous losses of individuals without losing the organizational structure that allows it to recover. These were not adaptations for surviving mass extinctions, they were adaptations for surviving ordinary ecological pressure day to day over millions of years. The mass extinction simply revealed how deep the resilience ran, how thoroughly the ant had built itself for a world that could at any moment change completely. Ancient Egyptian texts make relatively few direct references to ants, but the references that exist are precise enough to suggest close observation rather than casual acquaintance. The hieroglyphic symbol for the ant, a small, clearly segmented figure with 6 legs and prominent antennae, appears in contexts associated with industriousness and collective labor, occasionally as a determinative in words related to organized work. Egyptian grain storage facilities were engineered with features that modern entomologists recognize as ant deterrents: raised granary floors, specific plastering compounds, storage vessel geometries, suggesting that the practical challenge of ant intrusion into food stores was well understood and systematically addressed. More intriguing are references in some papyri to ants as indicators of underground water, a piece of observational ecology that aligns with the documented sensitivity of certain ant species to soil moisture gradients. Whether Egyptian farmers used ant nesting locations as a practical guide to water availability, or whether this was more generalized symbolic association, scholars still debate. What the record suggests is that the people of the Nile Valley watched ants with sufficient attention over sufficient time to accumulate genuine knowledge of their behavior. Knowledge embedded not in scientific texts, but in practical first, architecture, agricultural management, and the quiet, persistent observation of a civilization that understood better than most what it meant to build something meant to last. The ant appears in Greek and Roman literature with a frequency and specificity that reflects genuine cultural attention. Aesop's fable of the ant and the grasshopper, one of the most reproduced moral narratives in Western literature, is built on accurate behavioral observation. Ants do store food for winter, and the contrast between colonial foresight and individual improvidence that the fable exploits was not invented by a storyteller, but borrowed from a real behavioral difference visible to anyone who watched a garden through the problematic seasons. Aristotle described ant behavior in Historia Animalium with notable accuracy, correctly identifying their social structure, their food storage practices, and their treatment of the dead. Roman writers, including Pliny the Elder, documented ant agriculture, the harvesting and storage of seeds, and noted the social complexity of ant colonies with a directness that suggests first-hand observation rather than inherited folklore. Military writers drew on ant colony behavior as a metaphor for effective collective organization. The Roman legion's capacity for coordinated action without individual heroism was compared explicitly to the ant's way of working by several authors. The comparison was not meant as diminishment. In a culture that valued disciplined collective action above almost any other military virtue, being compared to an ant colony was a form of praise. The ant in the ancient Mediterranean world was a model, a small one, but a serious one. Across cultures that have lived in close proximity to ant-rich ecosystems, ant behavior has served as a practical environmental sensing system, a body of ecological knowledge accumulated through generations of observation and encoded in practices, stories, and agricultural timing. In parts of West Africa, the movement of driver ant columns, Doralus species, has been used as an indicator of coming rain. The ants seal their nest entrances and move brood to higher ground before major rainfall events responding to barometric pressure changes. Soil moisture shifts that precede the rain by hours. Farmers in several regions have used this behavior as a planting and harvest timing guide. In the Amazon, indigenous communities have long recognized that the presence of Azteca ants on cecropia trees indicates a healthy defended plant knowledge that guides harvesting decisions and forest management. In parts of South and Southeast Asia, weaver ant nests on agricultural trees are deliberately preserved as biological pest control, a practice that predates the formal concept of integrated pest management by centuries, and that contemporary agricultural scientists have validated in controlled trials as genuinely effective. What these knowledge systems share is the recognition that ant behavior is not random, that it responds to environmental conditions with a regularity reliable enough to plan around. The science and the indigenous observation arrived at the same conclusion from different directions. The ants were never not telling anyone who was willing to watch. In medieval European alchemy and natural philosophy, the ant occupied a symbolic position that reflected both accurate observation and considerable projection. Alchemical texts frequently cited the ant as an exemplar of industria, the virtue of purposeful, unceasing labor, and used the colony as an analogy for the ideal human community, a society in which every member knows its role, performs it without complaint, and subordinates individual desire to collective purpose. The ant's apparent ability to predict weather, to store provisions against future need, and to coordinate complex tasks without visible leadership made it for medieval natural philosophers a demonstration of natural reason evidence that divine intelligence was embedded in even the smallest creatures. Some alchemical texts attributed to ants a specific knowledge of underground minerals, proposing that the materials ants excavated in their tunneling, particularly in regions where anthills were found near ore deposits, could be analyzed to identify mineral wealth below. This idea, however fanciful in its theoretical framing, may have had some empirical basis. Certain ant species do preferentially nest in soils with specific mineral compositions and their surface mound material reflects the geology below. The medieval observer who noticed that anthills and metal deposits sometimes co-occurred was making a real observation if an imprecise inference. The ant, as always, was doing something specific. The human watching was doing the best they could with what they had, which is perhaps all any of us can say for ourselves. Edward Osborne Wilson, who died in December 2021 at the age of 92, spent more than 60 years making the case that ants deserve to be taken seriously, not merely as biological curiosities, but as a lens through which to connect and understand the deepest questions about the evolution of social behavior, the nature of altruism, and the organizing principles of complex life. His 1,990 book, The Ants, co-authored with Bert Holdobler, runs to 732 pages, weighs nearly 4 kilograms, about as much as a healthy newborn, and won the Pulitzer Prize for general non-fiction. It remains the most comprehensive single reference on ant biology ever produced. Wilson's broader theoretical contributions, sociobiology, the concept of biophilia, the superorganism framework, generated controversy that followed him for decades, particularly his arguments that social behavior in humans, like social behavior in ants, had evolutionary roots that biology could help illuminate. His critics were numerous and sometimes fierce. His response was typically to return to the data, to the field, to the ants themselves, which he continued studying into his late 80s. He described lying on the ground as a child in Alabama, pressing his face close to an ant trail, and feeling that he had discovered a world complete, complex, and indifferent to his presence, operating at a scale he had never imagined. He spent the rest of his life trying to describe what he saw there. Most people, he believed, simply had not looked closely enough. He may have been right. Human agriculture began approximately 10,000 years ago in the Fertile Crescent, the domestication of wheat, barley, and legumes by communities that had previously relied on hunting and gathering. This is considered one of the most transformative innovations in human history, the foundation of permanent settlement, surplus food, specialized labor, and eventually civilization as the word is commonly used. Leafcutter ants began farming approximately 50 million years ago. The gap between those two dates, 49,990,000 years, is long enough that the entire history of human civilization from the first agricultural settlement to the present moment fits inside it, without touching either end. The structural parallels between the two agricultural systems are not superficial. Both involve cultivation of a single domesticated crop. Both require soil preparation, waste management, pest control, and climate regulation of the growing environment. Both produce surplus that the colony or community lives on, rather than consuming immediately. Both have generated a degree of mutual dependence between cultivator and crop that makes separation fatal for both parties. The difference is that the ant's agricultural system has never failed catastrophically, has never required reinvention, and has never been replaced by a more efficient alternative. It has simply continued underground in the dark for 50 million years, producing food with a consistency that no human agricultural civilization has yet matched across comparable time. The comparison is not meant to diminish human achievement. It is meant to place it honestly in time. Ants have been moving soil for at least 100 million years. The cumulative effect of that activity on the planet's terrestrial surface is difficult to overstate. A single leafcutter colony excavates between 30 and 40 tons of soil over its lifetime, bringing subsoil material to the surface and redistributing surface material downward along tunnel walls. Multiplied across millions of colonies, across tropical and subtropical regions, the aggregate soil displacement is geological in scale, comparable to, and in some regions, exceeding the soil-moving activity of earthworms, which have long held the popular reputation as the Earth's primary biological soil engineers. Ant tunneling increases soil porosity, improving water infiltration and reducing surface runoff. It mixes organic matter from surface layers into deeper soil horizons, accelerating decomposition and nutrient cycling. It creates channels that roots follow, altering plant growth patterns across entire landscapes. In arid regions where earthworm activity is limited by low moisture, Ants are often the primary biological force shaping soil structure. Researchers studying long-term soil profiles in ant-rich environments have found measurable differences in soil chemistry, texture, and microbial community composition within and outside of historical ant territory, differences that persist for decades after the colony that created them has gone. The ants that built those tunnels are dust. The soil remembers them anyway, structured still by the labor of animals that have not existed for a generation, carrying the shape of a civilization that left no other monument behind. Mimicry, the dispersal of seeds by ants, is one of the most widespread and ecologically consequential plant-animal relationships on Earth. Approximately 9,000 plant species across every continent except Antarctica produce seeds with a nutrient-rich fatty appendage called an eleosome, specifically shaped and chemically composed to attract ants. Workers carry these seeds to the nest, remove and consume the eleosome, which contains lipids and proteins, calibrated to ant nutritional needs, and discard the intact seed in the nest's midden or a nearby underground chamber. The seed germinates in soil that has been loosened by tunneling enriched by organic waste, and positioned at a depth and location that often provides superior growing conditions to the site where the seed originally fell. The plant pays the ant in food. The ant plants the seed. Neither party negotiates. In the forests of the eastern United States, more than 30% of the herbaceous wildflower species Trillium bloodroot wild ginger, Hepatica, depend on ant dispersal for the majority of their seed movement. Remove the ants, and these plants' ranges contract. Their genetic diversity declines as seeds accumulate near parent plants rather than spreading across the landscape. The wildflower gardens of temperate forests were not designed by the flowers or by the ants or by anyone at all. They were assembled over thousands of years by the cumulative effect of millions of small transactions between a hungry worker and a patient seed, each one insignificant, and all of them together a forest. The role of ants in global carbon cycling is a relatively recent area of research, and the findings are still being integrated into broader models of terrestrial biogeochemistry. The mechanism is not exotic. Ants accelerate decomposition by fragmenting organic material, increasing its surface area and exposure to microbial activity. They move carbon-rich material between soil layers at rates that alter the depth and timing of decomposition. Their nests create localized hotspots of microbial activity, measurably higher rates of CO2 production than surrounding soil, because the warm, moist, nutrient-rich conditions inside an active nest are optimal for the decomposer communities that process organic carbon into atmospheric gases. At a local scale, these effects are well documented. At a global scale, they remain difficult to quantify precisely because the diversity of ant species, nest types, and ecological contexts makes generalization unreliable. What researchers have established is that ant activity is not carbon neutral. It accelerates the turnover of organic carbon from soil to atmosphere in ways that interact with climate variables, including temperature and moisture. As global temperatures rise and ant ranges shift poleward and upward in elevation, the geographic distribution of these effects is changing. The ants are responding to a warming world by moving into new territory, and as they move, they bring their biogeochemical influence with them, reshaping the carbon dynamics of ecosystems that have not previously contained them. The planet's carbon cycle has always had ants in it. It is learning now to have them somewhere new. The total biomass of all ants on Earth has been estimated in a 2022 study published in the Proceedings of the National Academy of Sciences at approximately 20 quadrillion individuals, 20 million billion ants. Their combined carbon mass was calculated at approximately 12 megatons, which exceeds the combined carbon mass of all wild birds and wild mammals on Earth combined. That number deserves a moment. Every elephant, every whale, every wild horse, every wolf, every wild bird of every species, weighed together, the ants outweigh them all. They represent, by carbon mass, roughly 20% of total human biomass, which is itself already the dominant animal biomass on the planet. The calculation methodology has been critiqued. Estimating total global ant populations requires extrapolation from sampling data across vast and poorly surveyed regions, and the uncertainty ranges are wide. But even the lower bounds of the estimate produce numbers that fundamentally reframe the question of which animals are doing the most biological work on this planet. By almost any measure of ecological activity, soil disturbance, seed dispersal, predation of invertebrates, nutrient cycling, the ants' contribution scales with their biomass. They are not a footnote to the Earth's ecology. They are measured by the weight of living carbon committed to the enterprise among its primary authors, small beyond individual notice, and collectively beyond individual comprehension, which is perhaps the most ant-like thing about them. Urban planners and systems theorists have returned repeatedly to the ant colony as a model for thinking about how cities function, not because cities are designed like ant colonies, but because both systems solve similar problems at similar scales using similar organizational principles. Both must distribute resources across a large, spatially extended population. Both must manage waste at the periphery without contaminating the productive center. Both develop specialized zones, residential, industrial, agricultural, defensive, that emerge from local behaviors rather than central planning. Both regulate traffic flow through reinforcement mechanisms. Roads that carry more traffic get wider and better maintained. Trails that carry more ants receive more pheromone and are followed more heavily. Both face the challenge of maintaining function despite the continuous loss and replacement of individual components. Workers die, residents move, and the city or colony continues without interruption. The urban theorist Jane Jacobs, writing in the 1960s about the emergent order of functioning cities, described principles of decentralized, locally driven organization that read in retrospect as a precise description of ant colony dynamics, despite the fact that Jacobs had no particular interest in ants. The convergence is not coincidental. Both systems are solving the same class of problem: how to maintain coherent collective function across a large distributed, heterogeneous population without a controller who can see the whole. The ant colony found one answer, the city found another. They are more similar than either would likely prefer to acknowledge. The Argentine ant supercolony that stretches 6,000 kilometers along the Mediterranean coast presents a philosophical problem as much as a biological one. If colony identity is defined by mutual recognition and cooperative behavior, if a colony is the set of individuals that treat each other as nestmates, then the Mediterranean supercolony is by that definition a single colony. It is also by that definition the largest animal society on Earth, a cooperative social unit spanning multiple countries containing billions of individuals, maintaining a shared chemical identity across a geographic range larger than the continental United States. Whether it constitutes a single organism in any meaningful sense, whether the superorganism framework applies at this scale, is a question that researchers approach with considerable caution because the answer depends entirely on how the key terms are defined and the definitions are not settled. What the thought experiment clarifies is something about the relationship between scale and identity. A human nation is a social unit held together by shared language, law, history, and institution structures that require conscious maintenance. The ant supercolony is held together by a shared chemical signature that no individual ant chose, maintains, or is even aware of. The coherence is real. The mechanism is molecular. The result is a society larger than most nations, with no government, no memory, no shared history, and no awareness of its own existence, persisting nonetheless as a unified social entity across distances that would take a human weeks to walk, maintained by nothing more than the chemistry of recognition, and nothing less than 100 million years of evolution that found in that chemistry something worth keeping. The venom of several ant species has attracted serious pharmaceutical attention, not as a curiosity, but as a source of bioactive compounds with properties that existing drugs do not replicate. Ponerotoxin, the peptide responsible for the bullet ant's extraordinary sting, acts on sodium ion channels in nerve cells, the same channels targeted by several classes of painkillers and neurological drugs. Researchers studying ponerotoxin's mechanism have identified structural features that may allow the development of compounds with more selective channel blocking activity than current drugs, potentially reducing side effects in pain management applications. Solenopsin, a compound from fire ant venom, has shown activity against several cancer cell lines in laboratory conditions, inhibiting tumor angiogenesis, the development of blood vessels that feed tumor growth at concentrations achievable without systemic toxicity in animal models. Formic acid produced by many ant species, and historically the source of the family name Formicidae, has antimicrobial properties that have been applied in various forms since medieval apothecaries dissolved ant bodies in alcohol and used the extract as a topical treatment. None of these compounds is currently in clinical use as an ant-derived pharmaceutical. The path from laboratory activity to clinical drug is long, expensive, and uncertain, and most promising compounds fail somewhere along it. But the research pipeline exists, and it exists because the ants, in the course of defending themselves from threats that existed long before humans did, developed chemical tools with a specificity and potency that pharmaceutical chemistry is still learning to match. Global warming is shifting ant distributions in ways that researchers are only beginning to document systematically. In temperate regions, ant species that were historically confined to warmer southern areas are extending their ranges northward and upward in elevation as previously unsuitable thermal environments become hospitable. In tropical regions, where many ant species already live near the upper limits of their thermal tolerance, warming is compressing viable habitat toward higher elevations and cooler microclimates, a process that ecologists describe as a thermal squeeze, in which the lower boundary of suitable habitat shifts upward faster than species can track it. The ecological consequences are not limited to the ants themselves. Changes in ant distribution alter seed dispersal patterns, soil structure, and the availability of ant-mediated services, pest control, pollination, facilitation, decomposition for the plant and animal communities that depend on them. In some regions, invasive ant species, Argentine ants, fire ants, little fire ants, are expanding their ranges faster than native species under warming conditions because their supercolony structure and behavioral flexibility give them advantages in disturbed and thermally variable environments. The native ant communities they displace had co-evolved with local plant and animal species over thousands or millions of years. The relationships built into that co-evolution do not transfer to the invader. The ecosystem retains the ecological role ants are still present, but loses the specific relationships that made the native ant a functional partner rather than simply an occupant of a shared space. Approximately 30 ant species are currently listed as threatened or vulnerable on the IUCN Red List, though myrmecologists widely regard this as a significant undercount. Most ant species have never been assessed, and many are described. From only a handful of specimens collected at a single location decades ago, the species most acutely at risk tend to share a common profile: narrow geographic range, specialized habitat requirements, low colony density, and ecological dependence on a specific plant or prey species that is itself declining. The Sri Lankan relict Ant-aphid simoni, the sole surviving species in a subfamily that was once globally distributed, is considered critically endangered, surviving only in a few patches of lowland rainforest. Several Australian ant species of the genus Nothamyomycia, the dinosaur ant, regarded by myrmecologists as one of the most evolutionarily primitive living ants, persist in small populations in isolated eucalyptus woodlands. For these species, extinction is not a distant theoretical possibility. It is a near-term probability if the habitat fragments that sustain them are cleared, degraded, or isolated beyond the point of recovery. What makes ant extinction ecologically significant beyond the loss of the species themselves is the loss of the ecological relationships they represent: seed dispersal, mutualisms, plant protection partnerships, soil communities organized around their nest architecture, relationships built over millions of years that cannot be reassembled from surviving components once the ant that held them together is gone. There is a habit of thought in which human intelligence is treated as the reference point against which other forms of cognition are measured and found almost always to fall short. Ants cannot write. They cannot speak. They cannot reflect on their own existence or communicate abstract concepts or build a telescope. By every measure of intelligence designed by and for human cognition, the ant fails. But the ant can do things that human intelligence, for all its sophistication, has never replicated without the help of technology the ant does not need. It can navigate a featureless desert using the polarization of skylight and the count of its own footsteps. It can maintain a living agricultural system across 50 million years without a crop failure. It can build a climate-controlled underground city with no blueprint, no engineer, and no central authority. It can coordinate the behavior of 1 million individuals into a coherent response to any threat, any opportunity, any environmental shift, using nothing but chemistry and proximity and time. The question of what ants know is at its core a question about what knowledge is and what form it takes. Human knowledge is encoded in language, in books, in institutions, in the accumulated explicit record of what humans have figured out and written down. Ant knowledge is encoded in chemistry and behavior, in the architecture of colonies and the structure of trails, an implicit record carried in bodies and renewed in each generation, of everything the lineage has learned about surviving on this planet across 100 million years. It is a different kind of knowing. It has lasted considerably longer, and on a long enough timescale, longevity is the only measure of success that does not eventually need to be revised. A worker ant does not sleep the way a mammal sleeps, in a long, consolidated block surrendered fully to unconsciousness while the world continues without it. Instead, it sleeps in fragments so brief they are almost not sleep at all. 253 episodes per day, each lasting just over 1 minute, distributed across every hour of the 24-hour cycle, with no preference for darkness or stillness or any particular corner of the nest. The antennae drop, the legs slow, the body settles fractionally closer to whatever surface it rests on. And then a minute later the antennae rise again and the ant is back, not groggy, not slow, not carrying any visible residue of having been for 60 seconds somewhere else. The total daily sleep adds to less than 5 minutes. A human who slept 5 minutes in a day would not survive the week. The ant functions normally, forages accurately, responds to chemical signals with full sensitivity, and performs every task in its behavioral repertoire without apparent impairment. Whether this reflects a fundamentally different relationship between neural tissue and rest, whether insect neurons recover from activity on timescales simply unavailable to mammalian ones, is not yet understood. What the data shows is the behavior, a life lived almost entirely awake in hundreds of brief surrenders to something that resembles rest without ever fully becoming it, distributed so evenly across the day that the colony is never at any moment without workers who are fully present and ready. The ant rests. The colony does not. The queen sleeps differently. Where the worker's rest is fragmented into dozens of brief episodes distributed across every hour, the queen's sleep consolidates longer episodes, fewer interruptions, something that approaches in its structure the kind of sleep a mammal might recognize. Research on fire ant queens found average sleep episodes lasting 6 minutes, with total daily sleep approaching 9 hours, nearly 2,000 times the daily sleep of a worker in the same colony, sharing the same genome, living within meters of each other. The physiological reason for this difference is not fully established. The queen's metabolic demands are enormous: continuous egg production, maintenance of the chemical output that regulates the entire colony, sustaining a body that must remain functional for decades. Deep, consolidated sleep may be essential to the repair and hormonal cycling that sustains those demands. It may also reflect the simple fact that the queen, living deep within the most protected chamber of the nest, faces no predator pressure, performs no foraging fights, no battles, and has no need to maintain the hair-trigger wakefulness that worker survival requires. She has workers for that. She has always had workers for that. And so she sleeps in the dark at the center of everything, while the colony turns around her, tending her, feeding her, carrying her signals outward to every tunnel and chamber, and the whole vast apparatus of the colony's life continues. Sustained in part by the simple fact that somewhere at its center the queen is resting, the surface goes quiet at night. The foraging trails thin, the traffic slows, the chemical signals that draw workers outward lose their urgency as temperature drops, and the risks of nocturnal exposure increase. But the nest itself does not sleep. Inside, the full range of colony life continues without reference to the day-night cycle that governs the surface world above it. Brood is tended around the clock. Larvae moved between temperature zones as the nest's thermal gradient shifts. Eggs turned and licked pupae monitored for the chemical changes that signal imminent eclosion. Fungal gardens are maintained on a continuous schedule, with workers trimming, seeding, and harvesting in overlapping shifts that ensure no section of the garden is left unattended long enough for a competing mold to establish itself. Queens lay eggs through the night. Waste is moved. The living architecture of the bivouac in army ant species adjusts its density in response to overnight temperature changes, thickening its outer walls as the air cools, loosening them as the colony's metabolic heat accumulates inside. The nest does not experience night as a pause. It experiences it as a change in surface conditions, a shift that affects some workers' activity and leaves others entirely unaffected, because the work that sustains the colony has no natural stopping point, no shift's end, no closing hour. The colony is always in the middle of something. It has been in the middle of something for 100 million years. In temperate climates across most of North America, Europe, and Northern Asia, ant colonies face a seasonal challenge that tropical species do not winter. Temperatures drop below the threshold for insect muscle function. Food sources disappear. Surface activity becomes impossible. The colony's response is not death, but suspension, a state of dramatically reduced metabolic activity called winter dormancy, in which the colony clusters deep in the nest, below the frost line, and waits. Workers cease foraging weeks before temperatures become lethal, responding to shortening day length and declining soil temperature, with behavioral changes that begin the preparation, sealing entrance, tunnels moving, brood and food stores, deeper, clustering around the queen in a tight mass that conserves heat through shared body warmth. Metabolism slows to a fraction of its active rate. Ant colonies in dormancy consume their fat reserves at rates low enough to sustain them through months without foraging. The queen stops laying eggs. The colony's chemistry simplifies the complex pheromone environment of an active colony, contracts to the minimum signals needed to maintain cohesion and queen recognition through the long cold wait. In spring, warming soil temperatures and lengthening days trigger the reversal. Entrance tunnels are reopened. Workers emerge onto surfaces that may still carry patches of snow. Foraging begins, slowly at first, and then with accelerating urgency, as the colony's depleted reserves drive the season's first foragers outward into a world that is once again available. The colony did not experience winter, it simply waited for it to end. Diapause is a state of suspended development deeper than dormancy, more precisely regulated, and triggered not by temperature alone but by a specific hormonal response to environmental cues that indicate unfavorable conditions ahead. In ant species that undergo diapause, the trigger is typically a combination of shortening photoperiod and declining temperature, detected weeks before conditions become critical. Juvenile hormone levels drop. Developmental processes in larvae arrest at a specific stage. The larvae do not continue growing not because they lack food, but because their developmental program has been biochemically paused, a timed suspension waiting for the chemical signal that announces winter's end. Diapause allows the colony to synchronize larval development with seasonal resource availability. Larvae that would hatch into workers during winter when no food is available to raise them, for the season that can sustain them. The precision of this timing is ecologically critical. A colony whose larvae complete development too early before spring food sources are available faces starvation. One whose larvae develop too late misses the spring foraging window that provides energy for the summer's reproductive investment. Natural selection has calibrated the hormonal triggers for diapause with a precision that matches local seasonal conditions. Populations of the same species from different latitudes have measurably different diapause thresholds adjusted to the specific seasonality of their home environment. The colony has no calendar. It reads the light, measures the cold, and knows. In the only way knowing is available to it when to wait and when to begin. There are moments in the depth of winter dormancy in the hours before dawn on a cold autumn morning, in the brief stillness that follows a major disturbance when the colony has not yet reorganized its response when the colony approaches. Something like genuine quiet, Pheromone traffic thins to its minimum. The trophylactic network slows. Workers cluster rather than move their antennae still, their bodies in contact with nestmates in a proximity that serves no apparent functional purpose beyond warmth and presence. The chemical conversation of the colony does not cease entirely, it never does, but it simplifies contracts, carries less. The nest breathes more slowly. The fungal gardens grow a little less tended. The brood waits. In these moments, the colony is still the colony. All the structure, all the complexity, all the accumulated evolutionary history of its lineage, but it is the colony at rest. The way a city is still a city at 4 a.m. when the streets are empty, the lights are mostly out, and the whole apparatus of urban life is present but not at this moment in active use. The comparison is imperfect, as all comparisons to cities are, but it captures something true, that even the most continuously active biological system has a register of quietness, a mode in which it maintains itself without extending itself. It holds what it has without reaching for more, and rests not, because the world has stopped demanding things of it, but because the resources available at this moment are sufficient only for maintenance, and maintenance done well is enough. In 2023, researchers studying the jumping ant, Harpegnathos saltator, the same species known for its remarkable Caste plasticity, observed something unexpected during the sleep phases of young virgin queens. The sleeping ants showed rapid antennal movements, twitching leg motions, and brief, involuntary postural adjustments that occurred during specific sleep phases and were absent during other sleep phases and during wakefulness. The pattern bears a structural resemblance to the REM sleep stage in mammals, the phase associated with dreaming, in that it occurs at regular intervals within the sleep cycle, involves motor activity inconsistent with the ant's resting posture, and appears to represent a neurologically distinct state from both wakefulness and quiet sleep. The researchers proposed cautiously that these movements might represent the motor outputs of an active neural process, possibly the replay of visual or behavioral experiences from wakefulness, a function that REM sleep is believed to serve. In mammals, whether ants experience anything during this state, whether there is something it is like to be a sleeping ant in this phase, is a question that neuroscience cannot currently answer, and may not be able to answer with available tools. The honest position is uncertainty. The behavior is real, the neural correlates are measurable, and what they represent in terms of inner experience, if they represent anything at all, remains genuinely unknown. Perhaps the ant dreams of running, perhaps it dreams of nothing. Perhaps the question of what it is like to be an ant is one that will remain permanently and productively open. The return of light and warmth to the soil surface does not wake a colony the way an alarm wakes a person all at once with a sharp transition from sleep to full activity. It wakes the colony the way a tide comes in gradually from the outside, moving inward from the most exposed workers toward the most protected ones, in a wave of reactivation that takes hours to complete and is self-amplifying at every stage. The first workers to respond to rising temperature are those nearest the surface in the uppermost tunnels where the soil warms earliest. These workers become active, begin moving, and in doing so generate pheromone signals and physical vibrations that travel downward through the nest, reaching workers in deeper chambers who were not themselves exposed to the warming stimulus. Those workers respond to their nestmates' activity rather than to the temperature directly, and their own reactivation sends further signals deeper still. By the time the sun has been up for an hour or two, the wave has reached the central chambers, the brood rooms, the queen's gallery, and the full colony is in motion. It is a cascade of local responses, each triggered by the one above it, traveling downward through the nest's architecture, like a signal moving through a network, which is of course exactly what it is. The colony wakes itself, one layer at a time, beginning with whoever is closest to the morning. Every evening, as light fades and temperature drops, and the colony's chemical signals shift toward the register of contraction and return, the last forager makes its way home. It is not designated as the last. It does not know it is the last. It is simply the one who has traveled farthest or found something late, or followed a trail that kept extending past the point where other foragers had already turned back. It carries what it found, a fragment of something, a droplet, a seed, and it moves toward the nest entrance by the same navigational mechanisms that have guided it all day. Polarized light stride, count, pheromone, gradient, landmark, recognition. The entrance when it arrives is partly sealed for the night workers inside, having already begun reducing the opening in response to the colony's shift toward dormancy. The last forager enters. The entrance closes further behind it. Inside, the colony receives what it brought, not ceremonially, not with any recognition of the lateness of the hour or the length of the journey, simply by Trophylaxis, the quiet mouth-to-mouth exchange that moves food and chemistry and identity through the colony without pause or punctuation. The last forager's contribution dissolves into the colony's collective stores. The individual who carried it becomes indistinguishable from the thousands of others clustered in the dark, and the nest settles toward its nighttime register, quieter, slower, its chemical conversations softened, carrying everything the day produced into the long, patient hours before the morning comes again, and the first worker nearest the surface stirs, and the whole vast, ancient, improbable enterprise begins once more. There is a version of this story that ends with a grand conclusion, a final claim about what ants mean, what lesson they teach, what human truth their civilization reflects back at us. That version would be easier to write, and it would be wrong. The ant does not mean anything. It is not trying to teach us patience or humility or the virtue of collective effort. It is not a parable. It is an animal pursuing survival with the tools evolution gave it, in the only way available to it across a time span so vast that the entire arc of human history fits inside it like a single afternoon. And yet, there is something that happens when you look at an ant colony with genuine attention, not the attention of someone identifying a pest or dismissing an inconvenience, but the attention of someone willing to follow a question wherever it leads. You find a system that has solved in its own register problems that human civilization is still working on: how to coordinate collective action without central authority, how to maintain infrastructure without a maintenance department, how to allocate labor without a manager, how to sustain an agricultural system across geological time without exhausting the land it depends on. The ant did not solve these problems the way a human would. It solved them the way evolution does, slowly, wastefully, through the elimination of everything that did not work across timescales that make human planning horizons look like a held breath. Is something that works. Something that has kept working through ice ages and extinctions and continental drift and every catastrophe the planet has managed to produce for 100 million years. The ant is not wise. It is not patient. It is not admirable in any way that requires intention. It is simply still here. And in the long, quiet arithmetic of survival, still here after everything is the only answer that has ever finally mattered. The end. Start, go to sleep, good night.